Dr. Abdur.
Hello.
Yes.
Yeah, can you? Hello. Hello. Good morning. Can you hear me?
Dr. Benji, you not seem good. Can you use your Dr. Abdur?
Yes. Can you hear me?
Yes, we hear you, but your camera is not on.
Sorry, I couldn't get you.
Yes.
Yeah, I couldn't hear your comments just now.
So which name you have joined in another system?
Yeah, right now, whatever you're communicating with, the same system.
So can you share your slide otherwise? We can.
Sure. Yeah, can you see my screen?
Yes.
Yeah, thank you.
Is that implicit, right?
Yeah, this is my presentation.
Yeah. So welcome you all for this Day-2 Indo-USA International Conference on Applications of Nanotechnology in Biology, Biotechnology, and Biopharmaceutics. So with this, we're gonna inaugurate our Day-2 session with the talk is done by the Dr. Abhilash Kangula from Kannan Nanomedicine Group, University of Missouri. He's going to give a talk on the whole nanoparticle-based urinary contact-free screening method for retinopathy of prematurity. So with that, I'd like to welcome Dr. Abhilash to give a share your slides.
Thank you for the introduction. Good morning, everyone. Respected faculty and guests and administrators and dear students of the Crescent University. First of all, thank you for providing this opportunity for us to share our research, present our research findings with the audience in the university. So today I'll be presenting our research, a part of our research that entitled Gold Nanoparticle Based Urinary Contact Free Screening Method for Retinopathy of Prematurity. So considering that most of the audience would be predominantly from the undergrad and graduate students, I would like to make this presentation as informative and educational for the students rather than showing a lot of data. My main focus is to introduce this research concept to the students so that they can benefit from this. So let us try to understand each of the terms that is stated in this title. So the most important term that you're seeing here is retinopathy of prematurity. So what is retinopathy of prematurity, or ROP, which is generally called? Basically, ROP is a retinal disorder that can lead to blindness in preterm babies. So the next question that comes is, what do you mean by a preterm baby? To understand that, let us understand first what is a full-term baby. As most of us know, a baby generally spends almost 39 to 40 weeks, around 9 months, in the mother's womb. So such baby which spends this many weeks before delivering out is considered a full-term baby, which is a healthy baby. Whereas in the case where sometimes due to complications during the pregnancy, a baby needs to be delivered much in advance before it completes its usual gestational period. In such cases, those babies are called preterm babies. So what is the consequence of being born as preterm? So the major problem with the preterm babies is that since generally the development of all the organs and tissues and the blood vessels develop usually when the baby is still present in the mother's womb, and the development goes on till the last week. Because the baby has come out early, the baby has incomplete growth of organs, tissues, or vasculature, which is not a healthy sign. For example, in this particular scenario of ROP, the retina of the eye, you know what is the retina is, like the deepest layer of our eye, which receives the light and enables us to see the world. So the retina of the eye has a vasculature or the blood vessels. Compared to a full-term baby, in a preterm baby that was born early, these blood vessels are not completely developed. Like what I mean is, if you see the picture, On the left-hand side, you can see a normal retina where from the center of the optical nerve disc, the blood vessels are starting and going till the periphery or circumference of the retina. Whereas in a retina of a preterm baby or premature baby, the blood vessels are growing from the center of the optical disc, but they have grown only till certain extent. They have not yet reached the periphery of the retina because the baby takes a few more weeks to develop these blood vessels completely till the periphery. So this is an example of where the blood vessels in the retina are not completely developed in a premature baby compared to a full-term baby. This is important because the blood vessels are the ones which carry the blood from the heart to all the organs, including the retina. And the blood also carries the oxygen and all the nutrients that are required for the metabolic activity of the retina. Since the blood vessels are not completely developed, the retina metabolism is affected. So now that we understood what the difference between a full-term baby and a preterm baby, and particularly with respect to the retina, it affects the development of the blood vessels. So generally the babies that are born before 31 weeks or the babies which are born less than 1500 grams in the body weight are generally are more preterm and are at a risk of developing ROP. So now let us understand now what exactly are the different stages of ROP and how it leads to blindness. So now that we have understood that ROP mainly develops because of the incomplete development of the blood vessels. As you can see in this picture, there is a clear demarcating line, which is indicated by this arrow. There is a white line that demarcates the area where there are blood vessels. It's called a vascular zone. And there is an area which is not developed with the blood vessels. It's called avascular zone. So this is called stage 1 ROP, where there's a demarcation line between these two zones where there is blood vessels and there are no blood vessels. And the stage 2 ROP is where this line is demarcating these two zones becomes thicker like a ridge. This thickness is because the blood vessels, instead of developing towards the periphery of the retina, they're developing along this line. This is called abnormal development of blood vessels, which makes this line to become thicker like a ridge. Both these stages of ROP 1 and 2 most often do not require any treatment, because most often, though this ridge forms, eventually our body recognizes this and tries to get back to normalcy by developing the blood vessels back to the periphery. But the thing is, like in this case, you can see the blood vessels are developing back to the periphery from stage 1 or stage 2. But the doctor always needs to screen these babies of stage 1 and stage 2 to make sure that the disease is not advanced further to the stage 3, where it becomes little bit of danger. The stage 3 is basically this ridge that's separating the vascular and avascular zone become much more thicker because of the development of more blood vessels along this line. Instead of the blood vessels developing towards the circumference, they're developing along this line, which makes the blood vessel, the ridge becoming thicker and thicker, and it starts coming upwards, and eventually it pulls out the retina from its place. That's called detachment of retina in the stage 4, where you see that retina is almost detached from the eye. which will eventually lead to blindness. So the stage 3 and 4 is where it becomes a little bit of concern, and then where we need to treat this abnormal development of blood vessels. Basically, the treatment consists of laser treatment to stop the growth of these abnormal blood vessels, and also uh, injection and administration of anti-VEGF injections to stop this blood vessel growth in an abnormal direction. So the summary of this slide is that the ROP starts with stage 1, where there is a demarcation between the vascular and avascular zone, and this demarcation keeps becoming thicker and thicker because of the development of blood vessels only in that demarcating line, eventually pulling out the retina. So how do we manage this disease is that the doctor need to continuously screen this preterm baby's retina to make sure that the blood vessels don't develop abnormally similar to the stage 3. Once the ophthalmologist sees the retina in stage 1 or 2, he should be able to start the treatment so that disease that does not further advance. So the main solution to ROP is the regular screening to prevent the disease progression to the stage 3 or 4, eventually leading to the blindness. So let us now know about some numbers regarding ROP and how prevalent is this disease in US and also in India. Coming to the US, basically there are around 4 million babies that are born every year, and out of which 15,000 babies are born preterm and then will develop ROP, which requires, and then among them 1,500 babies will require treatment, which means stage 3 or stage 4, and eventually 500 infants will become blind every year due to ROP. When it comes to India, these numbers almost multiply 10 times because of the population of India, and also because of the number of babies that are born preterm, and also due to the lack of facilities and the ophthalmologists similar to that of developing countries. So in India, annually 26 million babies take birth, out of which approximately 2 million are born preterm and are at risk of developing ROP. And among these people who are at risk, almost 38 to 51% develop ROP, which means that it's a very a dominant problem in countries like India and other developing countries, which has a huge population but not enough facilities and ophthalmologists to support the growing population of preterm babies. So what is the solution to the ROP? As I was telling in the previous slides, the treatment is not the solution to the ROP, because we need to prevent the progression of disease to a stage where treatment is not required. Basically, we need to catch the disease when it's very early. So we need to prevent the development of ROP beyond stage 1 and 2. As the age-old adage goes, prevention is better than cure. So for this to happen, screening is the solution for ROP, which means that retina of every preterm baby that is born needs to be screened once in every 1 or 3 weeks to check for any abnormality in development of blood vessels. Once we find any abnormality in development, the treatments to start immediately. And then till the blood vessels develop to the periphery of the retina, the screening needs to be continued, and then the baby can be discharged from the hospital. As you can see, the stage 1, stage 2, stage 3, and stage 4 we have seen in the previous slides. at the stage 1 and stage 2 itself, we need to prevent the progression to the further stages. For that to happen, screening is the solution. And so what is the current gold standard that is being used for screening ROP? The current standard that is being used is called an instrument called indirect ophthalmoscopy. It's basically an instrument, an optical instrument with a microscope, and then it has a bright light source and that shines on the eye of the preterm baby, and then an ophthalmologist which is trained in this procedure will have to observe the retina and its blood vessels. And what are the disadvantages of this procedure? So this is a very stressful and painful procedure for a preterm infant, which is already very fragile and weak and susceptible to many infections. As you can see in the pictures, the procedure needs that a sort of clip has to be put on the eyelids of the infant to be able to open the eye wide open. And they also put dilating drops to dilate the iris, and also they shine a very bright light, which is for a longer duration of time till the ophthalmologist can able to see the retinal blood vessels. So it's a very painful and stressful for the infant and also for the parents. And then there are research studies that shows that the stress and pain has long-term psychological and cardiovascular and gastrointestinal disorders on these patients, on these babies. Moreover, even the father and mother are also known to suffer from post-traumatic stress disorders. So that the main disadvantage of this screening procedure is pain and stress for the infants and for the parents. The second disadvantage is that this procedure needs to be done only by a trained ophthalmologist and in only specialized hospitals, which are very less accessible in low and middle income countries and also in remote locations. And the third limitation is that this procedure is very expensive and the instrumentation is also sophisticated and expensive. So there is a need to develop an alternative screening process. So in our research group, we thought that we will develop a kind of lateral flow immunoassay or a kind of a dipstick sensor, which is based on urine sample that is taken from the babies, and then we will measure the levels of a particular biomarker that indicates the presence of the ROP. So as we all are aware now, due to the pandemic in the last 2 years, most of us would have come across these lateral flow immunoassays or at home test strips, which are very convenient for us to use, and also they are very inexpensive. So we thought we will use the same concept to translate the same concept as a screening method for ROP. So the advantage of this method is that this is contact free, which means that we are no longer having we're no longer doing procedures that are painful to the babies. There is no contact with the baby during the procedure. What we need is only the urine sample, which we can collect using a cotton ball placed in the diaper of a baby. Yes.
We have only 5 minutes.
Okay.
Yeah.
Sure. Yeah. And then the presence of the biomarker in the urine sample will give us a test line, a red color test line, due to the presence of the gold nanoparticle in this strip. And then we are using a smartphone which has an app that can quantify this line intensity and give us the level of biomarker. And what is the biomarker which you are measuring here is 8-hydroxy-deoxyguanosine, which is an oxidative stress biomarker. And we hypothesize that the levels of this 8-OHDG will increase as the ROP stage advances. So this level of 8-hydroxyguanosine, which we are able to measure using this dipstick, is indirectly serving as a screening method for ROP. So now, if you go back to the title again, this is the gold nanoparticle based urinary contact free screening method for retinopathy of prematurity. So now that we have understood the concept of this, now I'll just share the results quickly because of lack of time. As you can see here, the test line, you can see a red color on the test line, and here is the concentration of the biomarker which we're detecting. As the concentration is increasing, the intensity of the line is decreasing. So this is an inverse relationship. And you can see here, stage 0, when there is no ROP, the level of this biomarker should be very low. So the color should be high. Similarly, when the stage is progressing, the color is decreasing, indicating that the biomarker levels are increasing. So in a way, we can say that this biomarker is serving as a good indicator of the stage of ROP. You can see on the right hand side a box plot which says that stage 0 patients' urine sample are showing the levels of this biomarker, which is around, on an average, 25 nanograms per ml, whereas stage 1 to 3 babies are showing almost twofold higher, which is around 50 nanograms per ml, which means that this biomarker is able to indirectly detect the stage of ROP. And then we are using this cutoff value of 25. When we compared this with the standard fundus examination or standard ophthalmoscopy, our sensor is able to detect the disease 85% of times. Sensitivity is 85%, which means that out of 100 babies which are showing ROP disease by the gold standard method, 85 of them are able to be detectable using our screening method, which is painless and inexpensive. So to conclude, sorry, ROP is a very devastating disease for the preterm babies, and its prevalence is increasing across the globe, including India. And timely screening of these babies is the only solution to manage the disease. The current standard procedure is very painful and expensive. So we have developed a painless smartphone-based dipstick assay, and then we are trying to further improve its sensitivity to make it 100%. And I would like to also acknowledge our team who has worked with us in this project. And thank you for this opportunity to share our research findings with the audience here. And I hope the students can benefit something from this. Thank you. And if you have any questions, I'll be glad to answer.
Thank you so much, Abnesh, for your presentation. Can the topic is open for discussion. Maybe 2 questions are allowed from both online and also campus. Yeah, any queries? Okay, I hope everyone understood well. Okay, thank you so much, Dr. Abnesh.
Thank you.
Thank you. Dr. Suresh, Manager Suresh.
Yeah, hi. I'm
Yes, please open your video.
just give me one minute. Can you see the presentation slide?
Yeah.
Okay.
Can you share your video here, participant?
Yeah.
What are you seeing? Your camera is turned on?
What is it?
Your camera is turned on?
Oh, I don't have a camera. I just have a microphone.
Okay, fine. Okay, the next presentation by Dr. Dhananjay Suresh from Crescent Nanotechnology Research Laboratory, Department of Radiology, University of Missouri. He will now deliver a talk on development of cancer nanomedicines to target the drug resistance mediated by AXL in lung cancer. Dr. Suresh, please take over the session. Thank you.
I appreciate for the kind words and the introduction. I would first like to thank the members of the Indo-US conference, organizers, distinguished guests, professors, and students for this great opportunity to share and discuss our research. My name is Dhananjay Suresh. I'm a senior researcher at the University of Missouri. And today I would like to share our latest research findings in the area of lung cancer therapeutics and cancer biology. Specifically today, we will talk about lung cancer, especially non-small cell lung cancer, which is responsible for very high mortality rates, and it has an overall 4 to 5 year survival time in patients. Therefore, there's a high clinical need for efficient therapeutics to combat lung cancer. So with that in mind, today we will talk about the development of cancer nanomedicines to target drug resistance mediated by AXL in lung cancer, where AXL is our target oncogene that belongs to a family of receptors known as receptor tyrosine kinases, also known as RTKs. So just to introduce RTKs, so RTKs could also be like growth receptors on cancer cells, and these lead to uncontrolled cell growth or proliferation by signaling to the downstream pathways within the cancer cell. So eventually it leads to proliferation of cells by uncontrolled growth, and finally metastasis, where the uncontrolled tumor cells can escape into the blood vessels and get transported to the various parts of the body, and the cancer spreads in that way. So introducing to the main RTKs. So in cancer cells, we have several RTKs such as c-Met, you have VEGF, you have EGFR, HER2 and HER3, and IGF. Out of which EGFR is a well-known receptor tyrosine kinase. It's a growth receptor, and it leads to uncontrolled growth in cancer cells. And because EGFR is commonly expressed in many cancers, most common therapies involve tyrosine kinase inhibitors, which are small molecular inhibitors which target EGFR. And for example, you have erlotinib, gefitinib, and osimertinib, which are currently used in clinics to target the EGFR. And as you can see in figure B, we see here that when a patient expresses high expression of receptor tyrosine kinases, the overall survival time decreases in patients. And therefore, there is a clinical need to target these EGFR-based signal. Now, coming to AXL. So what happened is initially when patients were being treated with TKI inhibitors, they found that within a year, the treatment would relapse and the treatment wouldn't work. And it took some time, but slow research, we figured out there is an unknown RTK known as AXL, which is causing signaling post-treatment with EGFR inhibitors. And we found that when AXL expression increases, as you can see here, when patients overexpress AXL, the survival time of those patients drastically decreases. And the overexpression or hyperactivation of AXL is seen in almost all patients post-treatment with EGFR inhibitors. AXL has also been implicated in proliferation, tumorigenesis, and metastasis in cancer. So it has a central role in drug resistance to TKIs. As you can see in the pathway, the AXL receptor has connections to the PI3-AKT downstream pathway, which goes to the mTOR, and this is required for cell proliferation, especially in cancer. So we expect that AXL controls the cancer proliferation pathways, which leads to survival of cancer cells. In order to combat AXL expression in cancer cells, what we designed is we wanted to use siRNA therapeutics against AXL. A quick introduction to siRNA. It's used for high specificity targeting. It involves a fragment of RNA, which is very specific, which incorporates the dicer, organoid, and RISC complex machinery within the cell, which is already present in all cells, in order to degrade the target gene. And by using siRNA, we can replace or increase the drug treatment efficacy. And because siRNA does not have side effects compared to pharmacological inhibitors. However, the main issue we face is that siRNA has a very short half-life in blood because it is easily degraded in environments. It cannot cross cell membrane by itself, is thermodynamically unstable, and especially it gets degraded in presence of serum. And as you can imagine, our blood has a lot of serum. So for example, in this image over here, which is a gel electrophoresis image, you can see that with time from 0 hours to 48 hours, we can see siRNA, which is shown over here, degrades. And at 2-hour mark, it already starts degrading by over 50%. Therefore, there's a need to have a design of a nanoparticle to protect siRNA in order for it to be delivered into the cell. So in this regard, our strategy was to develop targeted gelatin nanocarriers, which can be conjugated with the siRNA itself. So a brief description of our strategy is basically we'll make a nanoparticle, which is be biodegradable. It will be porous in order to serve to deliver agents. And we will be attaching it with an antibody on the surface. And this will target the overexpressed EGFR in cancer cells. And this will allow the nanoparticle to get into the cell and finally deliver the siRNA to target the AXL gene within the cell. So this is a brief schematic of the nanoparticle. We expect the gelatin is more of a polymer structure. So we expect the gelatin microporous structure and the antibody on the surface to protect the siRNA, which are short fragments attached to the surface of the nanoparticle. And this way, the AXL can be downregulated within the cell. So the first step for us to test our design was to understand whether the particles are stable. And for this purpose, we monitored the hydrodynamic size, which is the property of the nanoparticle within aqueous solutions. And we basically figured out that it is highly stable for extended duration. So this way, we figured out that our nanocarrier is first stable. Second, we wanted to know whether it actually protects siRNA. So as you can see here in the gel electrophoresis image, we see that the siRNA stays with the nanoparticle and it does not leach away in the gel, which shows that the siRNA is conjugated very specifically to the nanoparticle surface and it is protected by the structure of the gelatin and as well as the antibody. And as you can see here, even with time, the siRNA does not degrade drastically when in presence of the nanoparticle. So currently, technology in the literature shows that siRNA can be protected up to 12 hours. With our technology, we can protect it up to 48 hours. So next step, we wanted to study whether these nanoparticles can actually deliver siRNA within cells. As you can see here, this synthesis produces nanoparticles between, it's a range between 150 to 190 nanometers. And these gelatin nanoparticles are highly uniform and they're suitable for scale-up. The reason for scaling up is for eventual translation, we need to have a synthesis which can be scaled up very easily. Next, what we did is we took the siRNA and we labeled the 3' end of the siRNA with Cy5 dye. And what it does is because Cy5 is a fluorescent dye, we'll be able to monitor its uptake. And using this, we did in vitro studies, and you can see here in figure B, we see here that when free siRNA is used the sRNA is not able to penetrate the cells. But in presence of nanoparticles, you can see the pink signal, which is basically the Cy5 signal. You can see the Cy5 signal is shown in almost the cytoplasm of all the cancer cells in the image, which shows that the nanoparticles can deliver sRNA within the cells with no issues. Naturally, we wanted to know whether the sRNA is functional, whether it's actually downregulating the gene. So next, we studied the downregulation of the AXL gene using our carrier. As you can see here, sRNA is very specific. So this is just siAXL alone, which is transfected in cancer cells. And as you can see, as we increase the concentration, the AXL downregulation linearly decreases, which is the function of the sRNA. Next, we wanted to study what happens when we use our nanoparticles to deliver the sRNA. Here we have delivered the sRNA with and without the EGFR inhibitor, which is erlotinib, also mentioned here as ER. And we can see here that when we use these nanoparticles, the AXL gene completely gets downregulated. And we found the data to be highly significant. We have achieved about 75% to 80% downregulation with the current low concentration dose of gelatin nanoparticles. So with this, we wanted to study the sensitization of the current TKI inhibitors which are used in therapy. So in this case, for example, we are showing you here data for erlotinib, which is currently being used in clinics. As you can see here, this is the IC50, which basically shows the half inhibitory toxicity effect of the drug. And you can see here erlotinib has a toxicity shown by the black bars. And when we downregulate the AXL gene, and then we add the drug, we can see that the IC50 decreases, which shows that we can sensitize cells when we block AXL, which also indirectly proves that AXL drives drug resistance within the cancer cells. So this way, we used our nanoparticles, as you can see here, presented with the black bar, and we got significant data here, which shows that our nanoparticles were able to significantly decrease the IC50 by over twofold using the AXL. So with these results, we wanted to study the actual molecular biology of how AXL is able to control the cell proliferation as well as the metastatic behavior of cancer cells and cause drug resistance. So in order to do that, we did various studies. Over here, we are showing you some results from the Western blotting as well as the protein expression datasets as well as the apoptosis datasets. We have also done PCR and so on to study the pathways which are either associated with AXL or downstream of AXL pathway. As we can see here, when we downregulate AXL, whether we do it with AXL or with the constructs or with and without drug, what we notice first over here that the AXL gene gets downregulated in presence of sRNA, which is expected. The next thing we wanted to see is whether AXL, because AKT is required for cell proliferation, especially the phosphorylation of AKT, and it's downstream of AXL. We wanted to study what are the effects of downregulating the AXL gene. And what we found is that the moment we downregulate the AXL gene, the phospho-AKT levels immediately decrease within the cell. And they decrease even further when we use the drug in combination with our nanoparticles or AXL alone. And this shows that the AXL, or downregulating the AXL, is very efficient in curbing the cell proliferation pathways within the cell. So eventually we wanted to see further downstream whether it affects the phosphorylation of two proteins, specifically 4EBP1 and P70SHK. These two proteins are required for the function of mTOR. The mTOR protein is required for cell proliferation. And what we notice is when we use our treatments, the phospho-4EBP1 and the phospho-P70SHK also decreases within the cell, and they decrease especially when the drug is present. So this again proved to us that in combination with our drug, if we block AXL, we are able to curb the cell proliferation pathways within the cell. Next, we wanted to study what happens to the apoptosis rates, and we find that when we used our nanoparticles with the AXL in conjunction with erlotinib, which is the TKI drug, the late apoptosis rates increased over here compared to the SIXL plus ER or even drug alone, which shows that the apoptosis rates are responding with respect to the treatment of AXL downregulation. Finally, we also wanted to study what happens to the tumor suppressor genes when the AXL is blocked, because we expect AXL to be driving drug resistance and we expect AXL to be driving cell survival. So we wanted to study what happens. So for that reason, we studied the TP53 gene, which is a tumor suppressor gene in human mammalian cells, cancer cells. So what we found is when we downregulate the AXL gene, the P53 has a higher fold change expression within the cell. So an increased expression of P53 denotes that there's action of tumor suppression taking place within the cell. So overall, with this study, we could show that the behavior caused by AXL was completely shut down using our siRNA nanoparticles very effectively. So in the next slide, we're going to specifically look at how the AXL pathway controls the metastatic behavior within the cells. So in order to do this, we looked at proteins which cause metastasis, which are part of the EMT pathway, also known as the epithelial to mesenchymal transition stage. So this is where the cells, which are usually epithelial in nature, become mesenchymal. When they become mesenchymal, they become elongated and they become plastic. And this is how cancer cells can escape to the bloodstream. They become flexible. So we wanted to study whether downregulating AXL decreases the potential for the cells to become flexible, therefore stopping the metastatic behavior of the cancer cell. So as we can see here again, when we downregulate AXL with or without nanoparticle, especially in presence of drug, we see a downregulation of the expression of vimentin. Vimentin is very crucial for causing the EMT activity in cancer cells. The next, we wanted to study what happens to N-cadherin. N-cadherin is another protein which is part of the EMT pathway, and an increased expression of N-cadherin is seen in cancer cells. Again, what we see is when we downregulate it, especially in combination with the drug, we see a huge drastic reduction in N-cadherin. These results basically suggested that we could downregulate the activity of EMT if we can target AXL in combination with our TKI. Next, we wanted to study what happens to the decrease in what happens to the expression of MMPs, especially MMP2 and MMP9. These are two MMPs which are well known to be regulated by AXL, and MMPs are required for the transition of the cells to become migratory in nature, that is plasticity. So what we found is when we again did our treatments, we found a drastic reduction in MMP2 and MMP9 levels, which again proves to us, and these data was highly significant. So it proves to us that the downregulating AXL completely stops or curbs the metastatic behavior of the cancer cells involved. So with these studies, we use the expression data sets and use KEGG gene association analysis to find out whether there's an association of AXL with EMT factors, not only in the cell lines we studied, but also in all other cancer cell lines, especially in lung cancer. And what we miraculously found is that AXL had a very high correlation efficiency with other EMT factors in all other cell lines. Especially, we saw a high association of AXL with N-cadherin in almost all cancer cell lines we studied. We also studied the association of AXL with various pathways, and we found that AXL was actually having a very high association with P53, AMPK, P10, RAP1, MAPK, RAS, and chemokine pathways within the cancer cells. And what this data actually shows us is we can actually find out new biological markers within these pathways, such as the AMPK, P10, or MAPK or RAS pathways, and find out new biological markers to be targeted, which could be even more efficient than targeting AXL itself. So this could, and the rationale provided here in this study also shows that AXL can be a viable therapeutic target for combinatorial cancer therapies. So what I mean by combinatorial is essentially if you give the TKI inhibitors to patients and it causes drug resistance, if we can deliver the AXL siRNA along with the TKI drug, what will happen is the patient will never have drug resistance, because essentially we're combating the EGFR activity as well as AXL activity at the same time. So the simultaneous approach will improve the efficacy of the treatment in cancer patients. So with this, I would like to conclude this study. And so what we found in this study is AXL plays a crucial role in regulating cell proliferation and metastatic pathways. And using our strategy, we could downregulate AXL and shut down all the survival pathway mechanisms which AXL allows. And by downregulating AXL, the cancer cells became sensitized towards TKI such as erlotinib in our study, and therefore thwarting the drug resistance nature of AXL. We also show that gelatin nanocarriers were very effective in protecting siRNA, and siRNA treatment can be allowed this way to be highly specific because it has absolutely no side effects. The AXL siRNA will target only specifically the AXL gene. And using this technique, we can have combinatorial approaches to be strategized for several patients based on what kind of molecular biology landscape they have for their specific tumor, as we know that tumors are heterogeneous in nature. And based on this research, we are now working on unraveling several other resistance biomarkers, which can be used in conjunction or independent of the AXL gene. So with this, I would like to thank all of you for listening and helping us to share our research. And a special thanks to the Cancer Nanotechnology group at our university, which helped in finding these cancer biology and nanoparticle developments, especially Dr. Cannon, Dr. Anandi Upendran, and our funding from the Sharon and Buckstein Endowment Fund. And with this, I thank you all for listening, and I'll be happy to take any questions. Thank you.
Thank you for the Thank you for the wonderful lecture, sir. Any questions? Good morning, sir. Thank you for the wonderful talk. I have a question. Was this data same with, did it coincide with an in vivo study?
So currently we are working on the in vivo study, but our preliminary results show that downregulating AXL, especially in mice, decreased or sensitized the tumors in mice to the drug, the EGFR TKI.
So in that case, what would be your mode of delivery?
What would be, pardon, could you please repeat your question?
What would be your mode of delivery? Like, how will you be delivering the nanoparticle? Is it like IP or IC?
Yeah, so we are currently doing IP.
Sir, one more question. Like, why did you choose gelatin nanoparticle?
So
because
the reason
for more advanced polymeric nanoparticles, gelatin is relatively less stable when compared to other polymeric nanoparticles.
So yeah, so we actually previously we used to work with gold nanoparticles, and then we also tested liposomal particles as well as gelatin particles. The reason we chose gelatin is because we actually found higher stability of siRNA in presence of gelatin, because somehow the gelatin structure and the presence of antibody on the surface was helping siRNA to escape serum exposure. So which is required for siRNA to be more stable within the blood. And for this reason, we chose gelatin. And the next reason we chose gelatin is because the conjugation efficiency of siRNA to the gelatin surface is very high. The conjugation efficiency is about 90% or above. So basically, we can have very high loading rates of siRNA, which will allow us to decrease the dose of our final nanoparticle for the treatment.
Thank you, sir. Because gelatin has the RGD sequence, will it have side effects? Like, will it also harm the normal cells, the healthy cells?
Well, in our studies, we did not see any effects of gelatin nanoparticles at the current dosing we are doing in animals, as well as our in vitro studies.
Thank you, sir.
Yeah, thank you.
Sir, there are few questions in the chat box, sir. Why particularly use gelatin? Why not use other biopolymers? How to conjugate this gelatin with siRNA? What is the cross-linking agent used in this? And what is the polar present in gelatin? It is asked by Satya Rahman.
Yeah, so what we do is we take the gelatin nanoparticles and we activate the amine groups on the surface. And the amine groups are used, the primary amine groups on the gelatin surface, as well as the antibody is used then to conjugate with the siRNA, which has sulfur groups on its surface. So we use basically amine thiol chemistry in order to conjugate that. And with respect to the polar groups present on gelatin, well, the polar groups don't really affect the gelatin nanoparticles are very hydrophilic in nature, so we haven't had any issues with respect to the polar groups.
Another question, is there any positive treatment available for cancer, lung cancer, and which animal model can we use to avoid such nanotubes?
So currently we use, in terms of animal models, we use skid mice. We grow lung cancer tumors and usually subcutaneous tumors, and then we study it. And with respect to positive treatment available for lung cancer, so when it comes to lung cancer, it depends on each patient. So after the patient biopsy is done, we figure out the treatment regimen. So the treatment regimen goes as follows. We have first line treatments, which usually is immunotherapy these days. So we use immunotherapy agents to let the immune system fight against the cancer. If the first line fails, we usually go to a combination of chemotherapy along with the TKIs or tyrosine kinase inhibitors. So I hope I have answered those questions.
Okay, thank you so much, Dhananjay Suresh, for your wonderful time and spend for us for delivering your talk.
Thank you so much for this great opportunity for us to discuss and share our research as well. And I thank all the conference organizers and members and distinguished scholars and guests and students. Thank you so much.
Thank you. So, okay, participants, I will both in online and offline. We have on invited talk on the campus that will be delivered by Avinash Kumar from Indian Institute of Information Technology, Design and Manufacturing from India. So this session will be chaired by Dr. M.K. Shangita. So,
Introduction about him. Dr. Avinash Kumar is an assistant professor, Department of Mechanical Engineering, Indian Institute of Information Technology. He has completed his Bachelor of Engineering in Mechanical Engineering from Oriental Institute of Science and Technology, Bhopal, and MTech in Mechanical Engineering from IIT Kanpur. And he has also completed his PhD from IIT Delhi in Mechanical Engineering. He has also a work experience as a research associate in Robert Bosch Center for Cyber Physical System from IISc Bangalore. His research interests include laser machining, software engineering, micro and nano fabrications. He has also published n number of research papers in international journals and also book chapters. It is a great honor to have you here, sir.
So after this talk, we will have a tea break. Okay. Then there will be an oral presentation be held in this hall. So who are presenting oral and leftover last day early exceeded time, and today's scheduled presentation will be held in this hall. And all the online, even your friends or you are also presenting online, will be in the corresponding links that provide in the schedule. Okay. So if you have further question, please come to the registration. Not now, during the tea break.
So whatever you're saying is not clear, so it is echoing.
So the online, the information to the online participants. So after this talk, you need to go to your corresponding hall, that is OH 5 to 10, based on the participation code given.
So to join that OH, whatever it is, the link will be shared. It's a separate link for that.
Sorry, ma'am.
So to join the paper presentation in oral presentation in online mode, will a separate link be shared? Because there
Yes,
are
yes, a separate link shared in the program schedule.
Where will the link be shared, sir?
Last page.
Last page.
Please see completely that program sheet.
Program sheet. Sir, the program sheet what was shared was incomplete. Out of 280, only 261 PDF we have received. So we don't know the next of the PDF, and the links are also not in the PDF, sir. For the last batch, OH10.
Captain Budina, the session is going on. We are about to end the session.
Okay, sir.
I need to share. So just select the slide.
Okay.
Share. Okay, sir. Okay.
Yeah. Okay. So, very good morning to all. Myself, Dr. Avinash Kumar, and currently I am working at the IIITM, Kanchipura, and I am also affiliated to Stanford University as a visiting research professor. So, my basically I am a mechanical engineer. Mechanical engineer doing mathematical stuff in biology. Okay. So, I'm more aligned towards the product development. Okay. So if you see, today I am going to talk more about the texturing, micro texturing, and its application to biomedical devices. This is the outline of my today's presentation. If you see, so
any surface if you take, right, whether it's a metal, non-metal, or a composite, right, any metal can or material can be textured, right, using a various machining techniques, right? But we are choosing the laser texturing. Why? Because laser can machine any surface, right? Whether it's plastic or metal or it's composite or it's a tissue or anything, right? You can machine using the laser. Now, this if you see here, so these laser textured surfaces, right? We can design and we can make the texturing, and this texturing have a one advantage, right? These surfaces have a hydrophobicity, right? And this hydrophobicity have a great advantage in the field of a biomedical engineering, because these surfaces are highly antibacterial. Now, so what is the application of this kind of surfaces? These kind of surfaces can be used in a bioimplants, surgical tools, or they can also be used as in a microchannel, right? So, if we talk about the microchannels, in microchannels we have a friction, right? So, if you have a lab devices, you have a microfluidic devices, right? Their friction is friction is the main enemy, right? So we want to reduce the friction also. And here, when you talk about the nano devices or the micro devices, you have a problem of a mixing, right? Because is it audible? Yeah, okay. So I'm talking about this device, right? You can do mixing, even if you want to sort platelets, RBC, WBC from blood, right? Then based on the mass, right? Because they have a different mass, you can separate them also. And now, yes, laser. Okay. So first, I'll talk more about the texturing. Then I'll talk about the application. Right? So here, what we are going to use is a nanosecond pulse fiber laser, right? And this is the specification. All these things are the specification of the laser machine and the laser beam. So here we are going to use the laser beam of the wavelength 1064 nanometer, and the peak power which it can deliver is a 12 kilowatt, and the frequency we can vary between the 50 to 250 kilohertz, right? And if you see, the average power is 50 watt, and this spot, right, that beam dia is a 30 micron, and it can machine 10 by 10 centimeter of area, right? So now the various machining parameters, if you see, were the power, speed, frequency, and number of passes, and the various design parameters were the width. So if you have a texture, what should be the diameter, what should be the depth, and what should be the separation, right? This is very important. So based on the now here, if you see, so here we are explaining the mechanism of a laser machining. What it does, it will do two things, right? It will create a crater, right? A valley, right? Whenever a laser will interact with the material, right? So it will have a So what it will do, it will melt and vaporize the material. First thing, and second thing, what it does is it will do some chemical modification, right? Because of laser, this metal will undergo a very high temperature, right? And at very high temperature, it will react with the whatever the air or the gases there in the surrounding, right? And it will form some oxides, nitrites, or some other compounds, right? If you because in air we have mainly hydrogen, nitrogen, oxygen, so it will form either this hydride, nitrites, or the oxides, right? If you use some other gases, it will react with those gases and it will form the some other compounds. Now, so here what we are doing is we are designing the textures, right? So we have a designing parameters like depth. Yeah, here if you see, what should be the this is a texture, right? This blue color is a texture, and here we are taking the single pit or a texture, and so so it has a diameter d, and right, and this is the height actually. We can say d or the h. This is the height of the pit, and there's separation L. This L is the separation, right? So what we did, we applied the principle of energy minimization, right? And we get the what should be the dimension of this pit, so that whenever we put a water or a liquid on this surface, it will not penetrate, right? For that we need a Cassey-Webster state, right? So there are whenever you have a surface, solid surface, and when you put a liquid or any fluid over it, you have two conditions. One is the Wendell state, means liquid will penetrate into the cavity, and another is the Cassey-Webster. It will hang on the cavity and it will not penetrate, right? We want a Cassey-Webster state. So, and here the forces, if you see the forces, one force is the surface tension and another force is the pressure, applied pressure. If you have a surface and you are putting a liquid, you have a static pressure. When fluid is flowing, you have a dynamic pressure, right? And then at the interface, you have a surface tension. These are the two forces which are balancing, right? And here, so with that, we got what should be the dimension of this texture, and we fabricated using the laser machining. Here you see, this is the plane surface, and this is the textured surface, right? And on these two surfaces, we did the goniometry. And what we observed here on the plane surface, we had a 64 degree of the contact angle, right? Contact angle actually tells you about the wettability of the surface, right? How hydrophobic a surface is. Now here on the textured surfaces, only just making the dimples or making some modification, we are getting around the 148 degree of the contact angle. Whenever you put a droplet, it will like a lotus leaf, right? Whenever you put a liquid, it will just flow. Now, so these textures actually, if you see, these are the they are very time dependent, right? So here, what we are doing, we are taking a surface, right? Here we are taking the biomaterial, this titanium alloy, Ti6Al4b, and on that we are making the this kind of textures, and on the because this is mainly used in the bioimplants and surgical, right? So we are taking that, and when you do the analysis, time dependent analysis, then what we observe after machining, right? Day 0, after machining, we saw it's a very hydrophilic, right? And as number of day passes, it becomes the hydrophobic, and after 5 days, it becomes the super hydrophobic, and after that, hydrophobicity will not change, right? So if you see this this behavior, right? So the one question which will come in your mind is what is the reason behind it, right? So to investigate the reason, right? We did few analysis. Here, if you see, after machining this point, right? First day, at all the parameters, right? Whatever the machining parameter you use, whether it's power, frequency, or number of passes, everything, whatever you use, you will get the hydrophilic only. But after 5 days, at all the parameter, you have the hydrophobic samples, right? And this hydrophobicity, you can make it at your own will, right? So here, what we did the XRD analysis on these surfaces, right? So what XRD analysis will tell? It will tell you about if you have surface, what are the composition, right? What this surface composition. So if you see there are because we are using the titanium alloy, right? So the main thing will be the titanium, right? When it will react, it will form some oxides, nitride, right? So you will get maximum because we are using the Ti6Al4b. So titanium, aluminum, and vanadium will be there, right? And oxygen will also be there because of the oxidation of the surface. So what we observed, so we did after machining, pristine after machining, after saturation, and after we put 15 days in the water, and then we saw the what is the behavior or the how surface is behaving, right? So what we observed this, if you see this, the first black one is a pristine, then second row is a after machining, and third row is after saturation, and fourth one is after dipping in water for 15 days. So what we observed, or if you see here, right? So there is a increase in the oxygen content on the surface, right? So from 20, right, 20%, it is increasing to the 39%, right, after machining because of the oxidation. It is obvious. But after saturation, you are observing a 40%, right? 39 to 40, only 1% increase. But if you see, there is only 1% increase in the oxygen. But the contact angle, if you see, right, at 39, you are observing a contact angle of a 20 degree, right? But at 40, after saturation, you are observing a contact angle of 150, right? There is a drastic change in the hydrophobicity, but there is a very little change in the oxygen content, right? So to investigate further, right, what we did the XRD, right? Same with same sample after pristine, after machining, after saturation, and after dipping in water, right? So what we observed that here, yeah, that is a valid reason that after machining some oxides will form, but these oxides are unstable. They are hydrophilic but unstable. And after a few days, as number of days passes, this unstable oxides will convert into the stable oxides, and these are the hydrophobic, right? And after that, there is no change. This is the reason behind the hydrophobicity. And so here, if you see, so same after machining and after saturation, some peaks, right? They are increasing. This green, this is increasing. Some oxides are increasing, right? Why? Because there is continuous change in unstable to stable, right? And then what we did, so we can fabricate a texture, right, which will give you some contact angle, right? You can design and you can fabricate, right? If you want for 60 degree, or if you want 80 degree, or if you want 150 degree, you can design and you can fabricate. So we took from 60 to 150 degree, we took at a 5 to 10 degree contact angle variation, we took 8 samples, right? 4 samples, if you take this, there is a nomenclature, right? A1, A2, A3, A4. A is for hydrophilic and B is for hydrophobic, right? So because if contact angle is above 90, it is hydrophobic. If it is below 90, it is a hydrophilic. So we took 4 sample hydrophilic and 4 sample hydrophobic. Now we want to investigate what is the effect of this pit height and oxygen content and surface potential, right? Because there are a lot of parameters here. So, so we measured this surface potential using the KPFM. Oxygen content we measured using the EDX, and the 3D profilometry we used to measure the depth of the texture, right? And contact angle we measured using the goniometer. So what we observe here, when you increase, right, the surface potential, contact angle will decrease. And when you increase the oxygen content or a depth of a pit or a texture, the contact angle will increase. This is the trend. Now here we did the XRD with the same samples, right? So here you see this contact angle, right? Contact angle is 59, 65, 75, 80, 125, 136, 141, and 148, right? This is the contact angle what we are observing, right? And these are the corresponding to that contact angle. We have this much of a surface potential or oxygen content, and this is the depth of a pit, right? And on that sample, we are measuring the water, what is the surface composition, right? So we can see here. Now here the top, right, top 3 sample, you can see this is the hydrophilic, and bottom 4 are the hydrophobic, right? So you can see hydrophobic sample have a similar trend or a similar oxides. Hydrophilic samples have a similar oxides, right? Right? Can you see or not? These are all 8 samples are machined samples actually, right? So here what we observed, there are some oxides, titanium oxides, Ti2O3, right? formation of Ti2O3, depletion of TiO2, and the titanium nitride are the reason for the hydrophobicity. So now, in the beginning, I talked, right, that whenever you are doing a laser machining, you will have the two impact. One is the physical modification, means it is creating a valley, right? And second, it will do some chemical modification, right? The chemistry of the surface will change, right? So how much is the contact angle because of the surface chemistry, and how much is because of the physical modification, that we develop a mathematical model and we calculated. And we also calculated the stability of these textures, whether these textures are stable or unstable. Stable means if you put a liquid or water on the surface, after 5 days also it will be hanging only, right? It will not penetrate. So this is a stable sample. So now, yeah, this is a lot of mathematical steps here. So here what we observe, right? So this will tell you the okay. So if you are observing a 150-degree contact angle, because after machining you have a 150-degree of contact angle, right? So you have a plane material, right? It has a contact angle of 64, right? 64 degree, and you are observing after machining 150 degree, right? So some addition is because of the physical and some because of the chemistry, right? So it will tell you, okay, this much is because of the physical and this much is because of the chemistry, and whether this surface are the stable or unstable, that also it will tell. So we took the most hydrophobic and the most stable sample for its application in the biomedical devices. So if you talk about the application, right? So if you have a micro channel or lab on chip devices, right, you have a flow, right? You want to mix something, you want to flow something, or if you have an application of a drug delivery, right? So you want to control the flow, everything, right? So their drag is very high because we are dealing with the micro and the nano channels, right? So if you see, the friction is very, very high, right? So we can reduce the friction, right, by using this texturing, right? So here what we did, we made a micro channel. So to make a micro channel, we take the surfaces or the plates, and then we textured it. Here you can see this black, right? Black is the textured thing. Now by using the alignment pins, we and double-sided tape, we glue it, right? Then we have the because this is a very cheap and you can say very economical micro channel which we made, right? And so using the syringe pump, right? Here you can see this is the micro channel, and these are the pipings, right? And using the syringe pump, we are maintaining a constant flow rate, and we are measuring the pressure here, right? So the slope, right? If you plot, the slope will tell you about the friction, right, or drag. So this line, right, this is the theoretically obtained, and this is the experimentally obtained, this deltas, inverted deltas, and these squares are the experimentally observed, and this line is the theoretically obtained, right? So this black shows the how much drag reduction in the textured channels, right? And this red is on the plane channel, right? So here you can see this slope will tell you about the drag. So drag is less in the textured channels, right? Now, so the another example or application of these channels in a surgical tool, or if you are using bioimplants, or if you are using prosthetics or other applications, right? So there we have we can say we have a different expectation, right? If you have suppose if you are using a biomedical in biomedical, if you want to use a implant, right? So what you want antibacterial surfaces, right? It should be antibacterial, and other osseointegration should be very high or better osseointegration, right? So with these textured channels, we observed a very high osseointegration, right? And we observed a very high antibacterial effects. And when you are using it as a surgical tool, you want very similar, or the toxicity is also an important parameter, right? So we tested toxicity, osseointegration, then antibacterial growth and adhesion on these bacterial textured surfaces, and then blood coagulation, right? So what we observed that this blood coagulation is same as of the pristine surfaces because it's desirable. When you are doing a surgery or somebody is using as a surgical tool, you don't want blood to coagulate very fast because of the surgical tool, right? So it is that blood coagulation was the same. But if you see the toxicity was also not there because of the texturing. Another parameter is the antibacterial, but we are getting the additional advantage of the antibacterial, right? When we compared with the, so here you can see here on the pristine surface, right? So what we did, so this we took hydrophilic and hydrophobic textures, right? And we we took the P. aeruginosa and S. aureus, right? Because we want to test both gram positive and the gram negative bacteria. So what we did on the pristine surfaces, we did the adhesion and growth, right? So what we observed a very high value, right? You can see 11.77 into 10 to the power 6 CFU per mil, right? But on the texture A, we observed a little less, and on texture B, which is a hydrophobic, we observed very less, right? If you compare here to here, right, it's 0.52 compared to the 11, right? With the aeruginosa or S. aureus both, right? So there is a reduction in the bacterial adhesion in growth with this kind of the texturing, right? Micro texturing. Now, same analysis we did on the agar plate, right? So here with this gram negative bacteria P. aeruginosa, when we adhere and we did the growth on these kind of surfaces, what we observed, there is a very less, right, growth and adhesion on the this kind of the textured surfaces, right? After that, yeah, these are the some image of the whatever the bacteria will adhere, right? SEM image of those bacteria. And now after that we fabricated the microchannel which will do the drag reduction, which will do the mixing, right? Because here we have a because of texturing we have advantage of the flow anisotropy. That means if you have a, suppose if you have water and with impurity, right, you can separate the water one side and impurity on the other side. So you can do the separation, you can do the mixing, right, because of the anisotropy. And you can use these surfaces in bioimplants and surgical tools, right? So this is the conclusion of today's my talk. Right, these surface textures were designed and fabricated using a nanosecond pulse fiber laser, and the super hydrophobicity of the textured surfaces can be attributed to the physical and the chemical modification, right, which enables the stable entrapment of the air pockets. And these XRD peaks of the laser textured surfaces predict the transformation of the base metal into titanium oxides, right, and absence of a titanium nitride, right, which is consistent with the EDX results, right? And we observed a 30%, if you calculate, nearly 30% reduction in the drag we observed on these textured surfaces compared to the plane surfaces. And here a maximum reduction of the 96% to 98% reduction in the growth and adhesion we observed on the textured surfaces compared to the plane surfaces with the both gram positive and the gram negative bacteria. Yeah. Thank you.
So anybody would like to ask questions? Both online and offline participants?
Yeah, most oxides that were created after 5 days of incubation. So
oxides are created after machining. Whenever you will machine, right?
no,
Suddenly
after machining, the secondary oxides are
stable oxides.
So those are they naturally hydrophobic? Or?
Yeah, they are not naturally hydrophobic.
That was
Thank you for the talk, sir. So the titanium alloy that used after years, it could start corroding and like the surrounding tissue will get inflammation. So why did you choose titanium, sir?
See, in the beginning, I explained, right? With this laser machining, right? We can use any material, any biomaterial we can use to just to validate our whatever our concept, right? We use the titanium alloy because if you see mainly in maximum implants, right? We are using titanium alloy, so we do titanium alloy because of that. You can take even SS 316L also. We did same thing with this stainless steel and other biomaterials. So we got a similar.
So in our lab, we prepare the scaffold using NaO surface treatment. We used to use it for etching the polymer so that it creates roughness in the surface, and by that we increase the hydrophilicity of the scaffold. It also creates OH on the surface, so we increase the hydrophilicity by creating roughness or like it's a smooth surface, the polymer is smooth, and we create roughness by making by etching it with NaOH. Here we are doing something similar, like we are using laser and creating pits, but it's going the other way around. It is increasing the hydrophobicity and how it will react to the biological environment. So like if you put this in a stimulated body fluid or in the blend, what happens to the pit? Like will the cells go adhere in the pit? Will there be ions formed in the pit? Like
yeah, so same thing we did because all the this kind of structure what we made in IIT Delhi, right? IIT Delhi and IIT Kanpur, and we did these biological experiments in IIMS New Delhi, right? So there we tested with the stimulated body fluids. We tested, and what we found that same result. These surfaces are antibacterial, and the growth and adhesion will be very less on this surface. We found that. We published a paper also in 2018. So same thing with the stimulated fluids, body fluids, we did experiments.
anybody would like to ask a question? Thank you, sir.
Thank you.
I now request Dr. Hemalata, Professor in Dean School of Life Sciences, to honor our guest, Dr. Avinash Kumar.
Yes, thank you.
Thank you, ma'am. Thank you, sir.
Now we will have a short tea break. So the tea is arranged outside the hall. So you can go have a tea and come back. It's for 15 minutes. Then we will have the oral and poster session. Excuse me, we found a mobile phone. So if anyone left your phone, please come and get it.
Read out the participants today who are ready to present here. First, CPP 21 Bhuvneshwari, CPP 22 Rajalakshmi, CPP 23 Harini, CPP 24 Shraddha Saji, CPP 25 Vinit Kumar, CPP 26 Rajaga. I'm very sorry to pronounce your name wrongly. Anyway, CPP 27 Kavimani, CPP 28 Anish Ruban, CPP 29 Mohan Raj, CPP 30 Shruti, CPP 31 Archana Rajvel, CPP 32 Rishika Rai, CPP 33 Vikash, CPP 34 Prakash Krishna, CPP 35 Genevieve Maria Amrita. Right? This is the order. So please stick to your time, 5 minutes. Don't talk unnecessary introductions. Please go to share your results, highlight your results, and talk which is important. Right? Anyway, so the first candidate, Bhuvneshwari, please come forward. Priya. Priya is the first candidate. Please come forward.
In the properties, we can see it is a 3 properties. It is plastic adherence and surface, which expresses a surface marker, and a multipotent. It can differentiate into the adipocyte, which give rise to a fat cells, chondrocytes, which give rise to a cartilage cells, and osteoblast, which give rise to a bone cells. The placenta is a fetomaternal organ. Decidia, which is the maternal portion of the placenta. The other name for decidia is Decidia placentalis. My project work is Decidia bacillus, which is derived mesenchymal stem cells. Just isolating the placenta, which is taking the placenta from the C-section, and we can wash with the dalpicophosphate buffer saline and 0.25% trypsin EDTA. Along with that, we are adding the antibiotics, which no bacterial contamination, and filter with the nylon filter, and adding the RBC lysis buffer. The filter chunks are plated in the, which is seeded in the T75 class, that is DMEM medium. Okay, this medium is mainly for the isolation of mesenchymal stem cells. The first observed cells is the fibroblast. After several passage is nothing but the subculture. After the subculture, 2 to 10 subculture, the mesenchymal stem cells will be formed. This is the collecting the Decidia bacillus from the placenta and mixing and deserting and enzymatic digestion which separate the cells, plating the mesenchymal stem cells, and we are going for the preclinical animal study. Okay. And next is the enumeration of cell viability. We can do the cell viability process, proliferation, colony forming unit assay, scaling up, and cryopreserving. Cryopreserving for the future use. We can store the mesenchymal stem cells in liquid nitrogen for more than 10 years. After this, this is the colony which is formed. I have separated 5. Decidia bacillus 1, 2, Decidia bacillus 5. Next is the characteristic. The phenotypic characteristics is the flow cytometry. The cells were incubated with the mouse anti-human primary antibodies conjugated to fluorescence, which incubated and acquired by the flow cytometry. This is express the cell surface markers. The CD90, 105, 166, and 73, which express the cell surface marker. 133, 19, 14, 34, and HLA-DR, which lack of expression of mesenchymal stem cells. From this chart, you can easily understand. Next is the mesodermal differentiation. I have told already, mesenchymal stem cells which has capacity for 3 lineage. Osteogenic lineage, which is capable of forming bone cells. From these images, you can clearly understand. And adipogenic lineage, forming fat cells. This is the diagram. And chondrogenic lineage, capable of forming cartilage cells. These are done by the manufacture quick instructions. And next is the karyotyping of chromosome analysis. The metaphase chromosome analysis in the 10th passage. Passage is nothing but the subculturing of the Decidia bacillus derived mesenchymal stem cells observed to be normal. All the chromosomes analysis which will be normal. Next is a microbiological test, which is mainly for the detection of the bacterial endotoxin. Next is mainly the cytokine profiling of MSC. Cytokine is nothing but we are doing the 14 test. Okay, cytokine MSC which secretes cytokine which regulate as the immune cells. Okay, which is secreted by the immune cells, which is mainly for the wound healing process. It is used for therapy and regenerative medicine. Okay, mainly cytokine which is detected in the ELISA test. These are the 14 ELISA tests are performed in this procedure. This is the protocol for the ELISA test, and we do for the analysis which is in the statistical analysis, correlation, Wilcoxon sign rank test, and the t-test. And these are the in vitro studies, and again we go for the in vivo clinical animal study. For this animal study, Mr. albino rats are used. We have grouped 2 groups. One is that we have vehicle control, and second one is the test article, that is the mesenchymal stem cells are injected and observed for the 14 days, and we are done for the mortality, clinical signs, body weight, microscopy, histopathology.
What is the purpose of this study, madam?
So stem cells.
Why you injected this mesenchymal stem cells to the normal animals?
Animals, humans and the humans and the animals, both the biological system are same only, no sir. Just we are testing for wound healing process, sir. Wound healing. It is mainly for therapy, sir.
Did you create any?
Yeah, sir. Created in the rats, sir. Wound created, and after we this simply, sir. Yeah, sir. And next is the gene expression studies. 2 genes are expressed. Cyclooxygenase pathway.
Better you go to your results, madam.
Okay, sir.
You don't need these things. Better you cross your deadline.
So mainly this is for only therapy, sir.
Fine, fine.
Mesenchymal stem cells isolated from placenta can be used for therapy. Okay, more advantages are there and disadvantages is also there. Future application we can use for the heart, it can secrete all the cells like a heart cells, pancreas cells like that. We can MSC therapy, which is mainly used for COVID-19. Now the researches are going. Okay. These are the reference. Thank you.
Okay, thank you. No question. Please, next.
Can you briefly report on your novelties, treatments, novelty of your work?
Novelty, sir. I can use therapy, sir. It is mainly used in therapy. The research are going on for the COVID-19, sir.
No one reported this one?
No, sir. It is on the process, sir. Respiratory infection, it is on the process, sir.
Okay, thank you.
Okay, sir.
The next person, Bhuvaneshwari, research scholar, SRM College of Pharmacy. Rajyalakshmi, SRM College of Pharmacy. Harini, research scholar, Settlinad University.
It's not there.
So good morning everyone. I'm Harini, research scholar from Chettinad Academy. Today I'll be discussing on the topic of nanobodies as next generation toolkit in healthcare. So of course, if we talk about nanotechnology, then most probably it is going to be for a cancer development, since cancer always tops the list as it's a reason behind the increased death rates around the globe. So there are so many techniques that is available, but still people are seeking for a better therapeutic treatment strategy. And one among is monoclonal antibody treatment, which is really successful, but still there are so many limitations. So to tackle all these issues one at a time, of course, nanotechnology is going to be the only solution. And as you can see in the screen, there are a wide range of nanomaterials that can be chosen. So to select the best ideal candidate for our specific applications. So among all the chemically manufactured materials, nanobodies always is becoming a trend, because the nanobodies is nothing but an antibody, it's just a fragment of antibodies. So as you can see the structural aspect of antibody, the first one will be the conventional antibody, but this will be the heavy chain antibody, which is found specifically on certain organisms. This is called as a heavy chain antibody. Only in those antibody specific part, this is an variable domain, which is VHS, that is available in only heavy chain antibody, and this alone is what we call as nanobodies. So this was priorly isolated from camelid families of organisms. Later it was found that it can also be isolated from bacteria and fungi and many things. So comparing to the monoclonal antibody what we use for the treatment of cancer, the main problem with this is a molecular weight. It's very heavy actually. It will be around 150 kilodalton, and the stability of this particle will be determined by these two domains. So these two will work synergistically to give a stability for the whole particle. But whereas if we see in nanobody, we don't have such domains. It is just a small particle, but still it is so stable. Researchers are still working on how it is stable. But if we ask how, there is definitely no answer. But still, they are so stable. And if you see the characteristics, They have a high tolerance rate to the temperature, pressure, and pH, which is which they can which is very comfortable for us to use in a cancer since it is already always different than our normal cells. So these are some of the advantages and characteristics of nanobodies, and they can easily cross in physiological barriers like blood-brain barrier or blood CSF barrier, which is hard for a huge particle to cross because the size of nanobody is around 4 nanometers. Uh, so if you go for a chemically synthesized particles, if it is 1000 nanometers, what you synthesize, then we have to go for sonication. Then we have to filter to bring it to around 100 nanometer, which is the post-processing step is very hard. But in the case of nanobodies, like when you extract itself, it will be around 4 nanometers, which is very comfortable for us to work with. So regarding owing to their size, these are some of the advantages. They can deeply penetrate to the tumor tissues, and the drug release will be very specific, and they are long circulating. So these design theranostic approach, they can enhance immune response, and PDT is a photodynamic therapy. So where our peers, that is our photosensitizers, can be carried by the nanobody to a specific tumor location, and they can also act as a viral vectors. So I'm only concentrating on the theranostic application. So speaking on the diagnosis, they can act as a probe to detect the human epidermal growth factor receptor, which is overexpressed in the tumor tissues. And due to their rapid renal clearance, they will be a best candidate for us to use a non-invasive in vivo imaging. So in the therapy, the nanobody can be used in 3 setups. Like they can be used as such, so that will be naked nanobody, or they can be conjugated with targeting moieties and can directly target the tissue, or they can be encapsulated the drug and to deliver the drug at the specific site. So these are some of the nanobodies that are in clinical trials. As you can see, their applications is not limited only to the cancer, but they can also be used in other disease conditions. So to conclude, of course, nanobody take-home message will be, it is just an antibody, just a fragment of antibody, and there are so many researches going on in this. So that's its application is really wide. So these are some of the recent references people are working with, and this is the acknowledgement. Thank you. So, I'm working under Professor Agnishwar Giri Goswami.
Oh, okay. Good. So, what kind of adverse effect you can expect delivering the antibody as a sort of
nanobody? The only side effect is going to be biocompatibility, since it is not from a human. Of course, it. Yes, because it is already stable. It can tolerate heavy conditions and denaturance. So of course, biocompatibility will be the only issue. So if we go for a polymer encapsulation, that also can be overcome. So it's easy for us to work with.
Good. Thank you.
Thank you.
For antibody case, we can use primary antibody, secondary antibody, like that coating. So is it possible to do with the antibody?
Yes, sir. Anything can be coated. Their surface is like it's small, but the surface functionalization is very huge.
In case if you are taking antibody, it has a very high affinity with the receptor, similar affinity or identified.
Yes, sir. The affinity is really long because of the CD I've discussed already. Just a second.
What about the cost? Both are same.
It's affordable, sir. No, it's inexpensive compared to the monoclonal antibody treatment. You can see it has a CDR3 length is really great. So it's going to have a great affinity with the target antigens.
Okay, thank you so much.
Thank you.
The next person, Radha Sanjeev, research scholar at Rahman University. Please come first.
Next is Vinit Kumar. Please be there. Vinit Kumar.
Good afternoon to all. Myself, Shraddha Saji, Research Scholar, Department of Botany, under the guidance of Dr. N. Geeta. And my topic is large-scale activity of methanolic leaf extract of Artemisia absinthium encapsulated in chitin nanoparticles against Anopheles stephensi. Coming to the introduction. Artemisia absinthium, commonly called as wormwood, is a member of Asteraceae family. It is herbaceous perennial plant with fibrous root, straight, grooved and branched stem with silvery green appearance. It is native species of Europe and now it grows in different parts of North America, parts of Asia, Pakistan, Afghanistan and South America. Various parts of Artemisia absinthium contain higher amounts of secondary metabolites. It is a rich source of terpenes, phenolics, flavonoids, antioxidants and various biologically active compounds. From the ethnopharmacological point of view, Artemisia absinthium has various medicinal properties such as anti-handling, stomic, antimicrobial, anti-fertility, antipyretic, anti-tumor, anti-protozoal, anti-malarial, diuretic and insecticidal. And it's all about malaria. As per the World Malaria Report published in 6 December 2021, there were 241 million cases of malaria in 2020. And mosquito resistance is a currently used insecticides, and the emergence of development strains of provides the parasite has escalated the malaria problems in affected countries. Botanical insecticides may serve as suitable alternatives to synthetics because they have advantages such as being biodegradable, non-toxic to other organisms, eco-friendly, and have high specific activity against the targeted microorganisms. Coming to the life cycle of malarial parasite. Malarial parasite have two hosts, that is human and mosquito, and it completes its life cycle within this two hosts. And coming to the aim of my investigation, to investigate the in vitro larval potential of methanolic aerial part extract loaded chitin nanoparticles of Artemisia absinthium against Anopheles stephensi, and coming to its objectives: preparation of methanolic aerial part extract by cold maceration method. Next, encapsulation of methanolic extract using chitin nanoparticles. Characterization of methanolic extract. Characterization of methanolic extract encapsulated nanochitin by various spectroscopic methods, and study of in vitro drug release through different kinetic models. Evaluation of larval activity using encapsulated nanoparticles and toxicity analysis of encapsulated nanoparticles using zebrafish. A brief account of materials and method. First, collection and preparation of plant material. Then a plant extraction, that is aerial part, which I was using methanol. Then the preparation of chitin nanoparticles according to the procedure of Pani et al. 2019. Then encapsulation of the methanolic extract. Then entrapment efficiency of various concentrations of CNPS-ME. Then spectral characterization, which I was done, are UV visible spectroscopy, SEM, scanning electron microscopy, FTIR, X-ray, and theta potential analysis. Then the in vitro drug release kinetic models, that is zero order kinetics, followed by first order Hikuchi-Cosmer-Pepas model, and finding the correlation coefficient analysis. Then the mosquito larvicidal activity of encapsulated chitosan nanoparticles, and at last toxicity analysis using zebrafish model. And coming to the results of my study, first, entrapment efficiency of different concentration of Artemisia absinthium encapsulate methanolic extract encapsulated nanochitosan, and among the various concentrations, higher entrapment efficiency was found at 10 mg per ml, that is 86.45%. Then characterization study, that is UV visible characterization of drug encapsulated and its control is shown in this figure. Drug encapsulated and control were subjected to UV in order to absorb in order to understand their absorbing region, and all the two samples samples showed a peak at 250 nanometer. However, its absorbance value was observed for CNPs, higher absorbance for CNP MES, that is encapsulated one, compared to the control. Then FTR analysis. This is the table of functional groups which was observed in both encapsulated and control nanoparticles. And in case of encapsulated nanoparticles, there is three functional groups, that is alkylamine, CH group, and a halogen compound with various peak values is shown. Then the X-ray diffraction of CNPs and methanolic encapsulated CNPs. Methanolic encapsulated chitosan showed more number of peaks with high intensity compared to its control. Then this figure shows the zeta potential of control and encapsulated chitosan. And the encapsulated chitosan showed higher zeta potential value, that is positive 47 microvolt with millivolt, which ensures more stability of nanoparticles against agglomeration. And this graph shows the SEM micrograph of CNPs and CNP-ME. And as you all know, SEM image shows the morphology of both control and encapsulated at 50 μm scale bar. And SEM confirmed the formation of chitosan nanoparticles and encapsulated nanoparticles. The unloaded chitosan nanoparticles have a particle size of 140 nanometer, and the size of the methanolic extract loaded chitosan nanoparticle is of 260 nanometer. Hence, the size of the nanoparticle increases by addition of methanolic extract. Then this shows the figure of in vitro drug release kinetics. This table, I had done zero order kinetics, first order kinetics, Hikuchi model, and Cosmer-Pepas model. And this table shows the correlation coefficient and kinetic constant of different kinetic models for Artemisia absinthium encapsulated nanochitosan. And the model with the highest R-square value is considered as the best fit of in vitro drug release data. And in this case, chitosan CNPS ME, that is the encapsulated one, most suitable model was Hikuchi, having a correlation coefficient value of 0.78. And then coming to the mosquitocidal or larvicidal activity of Artemisia absinthium encapsulated nanochitosan, highest mortality rate was showed by encapsulated chitosan nanoparticle within 24 hours, that is 100% mortality.
Try to control, ma'am. Try to control.
Okay, ma'am. Last. Then coming to the last slide, that is the toxicity study using zebrafish embryo toxicity, which was done for 48 hours, 0 to 48 hours. Various features are shown in this figure, and no deformities are shown by the CNPS methanolic extract encapsulated nanoparticle. Coming to the summary, the point of present investigation was to blend effective chitosan nanoparticles loaded with Artemisia absinthium aerial part methanolic extract and evaluates its larvacidal potential against Anopheles stephensi. And based on the in vitro drug release percentage, R-square value and larvacidal activity of CNPs was subjected for toxicity analysis using.
Okay, now we understood your summary. I have only one question.
Okay,
see, this nanobot will have toxicity for larvae, right?
Yes.
But it is not toxic for zebrafish. How this miracle is happening? Then?
Sir, it shows toxicity.
You say it looks normal. After we treatment, zebrafish is hatched.
Sir, it is a short toxicity, so we can
How
use
toxicity is there?
below 10 micro.
That is the purpose, right? The purpose
Yes.
of the development of this nanocomposite is to, toxic to the larvae, right?
Yes, sir.
So that is the goal to achieve, right?
Yes, sir.
Then I don't think you need to check the toxicity again to zebrafish. Huh? I don't mislead you.
No, sir.
Anyway, thank you.
Okay, so during the question session, second person will come and upload your files. Next person will more, you come this side, madam. Sranda, Sasi, you come this side. So I have few questions. So why you have chosen root? You have taken the extract from root, right?
No, sir.
Sorry, aerial form. Okay. Then your zeta potential value is positive value. What is the reason you got positive value?
Sir, it shows the stability of the nanoparticle.
There is a chance to get negative value, right? But in your case, both the cases before, only 5% after attachment of things, both are shown positive value. What is the reason?
It shows the.
No, you have to check the structure of cytosine. So in the structure, amine group NH2 group will be there. When you dissolve it, it's become NH3 plus. So that is the reason you got the positive values. Okay. So more about this. Okay, good. Next person, Vinit Kumar. Vinit Kumar, sir. Vajakadari, research scholar, Centenary Applied of Research. Vajakadari. After Vajakadari, Kavimani is there, Guwahati University Research Scholar. Yeah, get ready. And you can go directly for your result and discussion.
Yes, sir. I just want to give some small introduction because to make understand.
The participants you can discuss during the answering.
Okay, thank you. Bro,
we are not able to provide the
answer.
Sorry.
Okay, thank you. Okay, sir. So, hello everyone. So, I never could have imagined that the bacteria can cure all the human diseases as well as even including cancer as well as neurological diseases. So, this makes our curiosity level become high. So, we are using a probiotic name called E. coli Nissl 1917 to reverse the acrylamide induced neurotoxicity in zebrafish model. So, this is our introduction part. So, neurotoxicity is nothing but it damages the nerve cells by natural or man-made toxic substances. So neurotoxicity is also cause of various neurological disorders such as schizophrenia, depression, as well as many neurodegenerative diseases. So next, acrylamide. So we have used acrylamide toxicity substances to induce neurotoxicity to the zebrafish model. So here is our probiotics. So probiotics are nothing but probiotic is a good bacteria to cure many diseases as well as E. coli Nissl 1917. So we have selected this particular strain to cure the diseases. So here is the novelty of this research. So E. coli 1917 has been studied extensively for its role and treatment of gastrointestinal comforts. However, the role is improving neurotoxicity has never been studied till now. The study is completing or paves the way in improving the treatment without any side effects. So for example, now the current treatment strategy is having more number of side effects. For example, like psychiatric disease, particularly called depression. So in depression, the people used to take a drug name called escitalopram. Escitalopram is the main side effect. It causes the obesity to the patients, those who are taking the drug. So our E. coli Nissel gives a proper efficacy without any side effects. So in aim to study the effect of probiotic E. coli Nissel on acrylamide induced neurotoxicity in zebrafish. So our objectives is, first we have checked the toxicity level of E. coli in the zebrafish model. Next we have checked what is the colonization period of zebrafish in the gut. The next one is to induce neurotoxicity using acrylamide. So for inducing neurotoxicity, we have used the acrylamide as a drug. So next one is ECN as a neurotoxicity. For acrylamide induced neurotoxicity, we have used the ECN model. Then is a methodology of research. So this is a structure of E. coli 1917, and we have induced the E. coli 1917 through the orally to the zebrafish. There is the animal imaging. So next slide, I will show you what is the survival rate of E. coli Nissel in the zebrafish gut. So in workflow 2, we have used larvae model as well as adult model. In larvae model, we have used like post fertilization period of after 72 hours. Then for adult model, we have taken 3 months older than zebrafish model. So this is a sample size. Then for the adult model, we have done behavior analysis, histopath analysis, and biochemical assays. For larvae model, we have done staining techniques. So these are our results. So for animal imaging, we have done 10 mole of ECN sample to the zebrafish. So this is the images, the survival rate of ECN in the zebrafish gut. And one more thing, ECN alone is not a fluorescent. So we have make a transformation by using M-cherry contained plasmid. So we have used a transformation technique. So here the ECN, here is the M-cherry. So we just use the transformation and do the research. So this is a day 1, day 2. So can you able to see that there is a fluorescence in the stomach part. So the stomach part contains a ECN. So if the days goes on, that little bit it's like in day 8, there is no presence of ECN in the gut. So the picture showing fluorescence of M-cherry tagged ECN. So the light filter will be like fluorescent. The intensity of fluorescence is high till day 5 and it slowly decreases. So this is confirms the presence of ECN in the gut. The next one is a behavioral results. For behavioral results, as I have told you, we have induced a neurotoxicity like acrylamide. So 1 millimolar acrylamide to the adult fish. So these are the differences we have seen in the adult zebrafish model. So we measured by using Anime software. So I just want to show you a small video. So this is a video of different controls. The control probiotic and acrylamide and probiotic plus acrylamide. So the thing is, I just play the video here. So if it's possible, you can able to see the differences. The acrylamide induced zebrafish showing it just stayed at the one part of the top of the corner of the like fish tank, and it's not moving anywhere. This is a probiotic plus acrylamide induced fish. So it improves the behavior of the zebrafish. So that means, and one more thing, after this we have done like histopath analysis as well as some other like biochemical analysis, even that shows no toxicity levels which is present in the probiotic as well as probiotic alone and probiotic plus acrylamide induced zebrafishes. So this is the histopathological analysis of zebrafish. So after the behavior analysis, we have isolated a brain from the zebrafish. So this is a picture of the brain. So the histopath analysis shows that the controlled probiotic and acrylamide treated and probiotic induced acrylamide treated fishes shows that acrylamide induced fishes shows there is a presence of gliosis, edema, as well as perivascular gliosis. It increases the size of the neuronal cells. As well as probiotic and acrylamide treated fishes shows that it reverses all the conditions, as well as there is no presence of cell death in the probiotic acrylamide treated fishes. Then this is a biochemical analysis. In biochemical analysis, we have done only the catalyst assay. So in catalyst assay, first we have done by using Bradford's method, we have estimated the protein. Then after that, we have done the cat assay. So in cat assay showing that there is an acrylamide treated fish, there is a presence of like the protein has been degraded, and the probiotic plus ACR shows that it improves the condition of the degradation of the protein. Next one, staining analysis. So the staining analysis has been done. We have used a stain name called acridine orange. So staining analysis only done in zebrafish larvae model. So you can see the control samples and probiotic. So just look at the arrow mark. There is a presence of like a neuronal cell death. So there is cell death presence in that. So the probiotic and acrylamide, the presence of cell death is there, but it's comparatively lesser than the acrylamide only treated on zebrafishes. So our idea is to improvise the treatment for the future directions, and we just want to eliminate the complete neurotoxicity for the human as well as few animals also having the same condition of neurotoxicity level. So these are the conclusions. The animal imaging showed a survival of ECN in the gut in the zebrafish model. So the novel tank test confirms that the acrylamide-induced neurotoxicity signifies between controlled, probiotic, and acclimated fishes. The histopathic result shows that that cell death has been removed in the probiotic plus ECN treated fishes. Then cat assay shows that the ROS has been measured and compared the controls as well as zebrafish treated fishes. Then hence it is concluded that the ECN plays a vital role in the protection and maintenance of neurotoxicity in the zebrafish model. So these pictures I just inspired from the small movie. So for example, like current now the various treatments are there, but still it has low efficacy and side effects. But probiotic is our future direction to treat many other diseases like neurological disorders, cancers, as well as even our novel coronavirus also. So thank you.
Where do you get this strain, sir?
Sorry.
The isolated strain, E. coli?
Actually, we have just bought the E. coli strain from the demo, like sir. Then we just cultured it.
Okay.
Cultured it. Yes, sir.
But go to your catalyst.
Just a minute, sir.
So you have shown catalyst was decreased in acrylamide-induced condition.
Yes, sir.
Why is that? You know what is the role of catalyst, right?
Yes, sir. The
Why is there decrease in
protein level, sir?
When you show the catalyst activity level is less, right?
Yes, sir.
In the acrylamide condition.
Yes, sir.
So normally is that the case?
No, sir. Actually, no, sir. Actually, here that's why I mentioned control also. In control as well as probiotic, the catalyst is very normal. There is not like a degradation of protein.
So why the catalyst is less? See,
Less.
Paras, see, when acidity stress is there,
Yes, sir.
the catalyst, when both the sodium and catalyst, they tend to release more to
Yes, sir.
carry the
Yes,
free
sir.
radicals, right?
Yes,
That
sir.
is the hypothesis.
Yes, sir.
But in your case, it is decreasing. Why
Yes,
is
sir.
it decreasing?
So yes, sir. For catalyst, as I said, we are using like hydrogen peroxide as well as glacial acetic acid. So what we're supposed to do is we just add the hydrogen peroxide for the certain level. So here I have mentioned like only how much protein has been degraded. So our protocol is like we have measured like in 570 nanometer in different time interval. 0 minute, 1 minute, 2 minute, 3 minute, and 4 minute. So what happens when you compare to the absorption level? 0 minute absorption is higher. When you compare to 4 minute absorption level, is completely lower. So that means that H2O2 activity is completely degraded when it's like acrylamide present in the particular brain sample.
So go back and check your results again.
Yes, sir.
Because I'm not fully convinced.
Okay, sir.
Thank you.
Yes.
Okay, thanks. Next I call Kavimani, Madurai University Research Fellow. Specifically, I have one question. So you have performed. Okay, sir, come please. You have performed ROS, right? So what about the value?
Values, for example, you asked me the absorption value.
Yes.
Absorption value is like the acrylamide induced, for example, it came at 0.278 initially for the 0 minute. And for the 1 minute for the 570 diameter, it was like 0.2, like 220, then 215, then 210. So when the time goes increases, then absorption level decreases. For that acclimated fish. For in case of controlled fishes as well as like the probiotic alone, then acrylamide plus probiotic treated fishes shows a different value than the acrylamide alone fishes.
Okay, thank you. Yeah, Kavimani, you can start now.
Good morning to all. I am Kavimani, research scholar. Good morning to all. I am Kavimani, research scholar, Department of Botany, Biotech University. My topic: in vitro anti-inflammatories and wound healing potential of synthetic coumarin encapsulatum cytosine nanoparticles. Introduction: Cytosine is a naturally hydrophilic which display many desired characters such as biodegradability, good biocompatibility, non-toxicity, and antimicrobial activities. The inflammation onset happen immediately after injuries or wound formation. The coumarin chemical known as 2H1 benzobiron 2,1, which consists of large class phenolic substance found in plants. Several studies have been indicated to coumarin nucleus as prospective candidate for development and anti-inflammatory and wound healing drugs. However, it applicable for topical use. This restrict to low hydrophilicity. It's hypothesized that if coumarin entrap in coumarin nanoparticles, objective conjugation and encapsulation of coumarin nanoparticles, cytosine nanoparticles, determinants of in vitro drug release percentage, those various kinetics models, evaluation of in vitro anti-inflammatories and wound healing potential, L929 cell lines of CNPS. Materials methods: synthesis of coumarin nanoparticles and preparation of coumarin encapsulated nanoparticles, conjugation or encapsulation of coumarin with cytosine nanoparticles, characterization of encapsulated nanoparticle UV, FTR, XRD, and SEM, determination of in vitro drug release kinetics models, 0, first order, AQC model, and Koppas model, evolution of in vitro and anti-inflammatory wound healing, the potential L929 cell lines of CNPS. The result: entrapment efficiency of coumarin encapsulation nanoparticles, different concentrations among the various of concentration 10, 20, 30, 40, 50 mg per ml. The entrapment efficiency was found to higher in 30 mg per ml, 91%. This in vitro drug release percentage of kinetics model, the 4 marks, 0 order kinetics, first order kinetics, EQC model, Koppas models. Correlation coefficients R squared derivative form of different kinetics models for coumarin encapsulated nanocytes. The highest values present in EQC models 0.97 UV. The absorbance peak of 250 nanometers. First source is the control source coumarin encapsulation. This FTAR, first one pick control, second is encapsulated nanoparticles. Another, the majority of functions, the FTAR control encapsulated nanoparticles. Among the majority of the functional groups of chemical compounds are almost similar to chitosan nanoparticles plus chitosan encapsulation nanoparticles. The fuse are different: sulfonic acid, anhydrate, hydrate, polysaccharide, sulfate absorption, halogen compounds are different. XRD analyze. Some control to compare in chitosan nanoparticles, the different 7 nanometers are highest value. So in vitro anti-inflammatory activities. The in vitro anti-inflammatory studies showed that CNPS process higher incubation activities for protein denaturation, proteinase, membrane deformation, hemolysis, hyaluronidases, fibrocellulose. The coumarin synthesis chitosan nanoparticles were assumed in scratch assays to determine its effective or splitting and migration activities of L929 fibroblast cells with different concentration 5, 10, 25, 50 each per ml. The absorption of 24 hours. The CNPS showed that the increase close, so 82% compared with chitosan. The first negative control. Second picture is coumarin encapsulated chitosan nanoparticles. Summary: the synthetic coumarin process are used to prepare the coumarin encapsulated nanoparticles. In the synthesis, coumarin encapsulated nanoparticles was subjected UV, FTR, XRD, SEM characterization, which confirmed the formation of nanochitosans. Among the variation concentrations of 30 ml per ml CNPS showed that highest entrapment efficiency by 91% and were subjected to various kinetics models are 0, first order EQC models and the Koppas model. The highest R squared values found in EQC models in 0.97% followed by Koppas model. The in vitro and anti-inflammatory studies showed the significant inhibition percentage of proteins denaturation. Yes, sir.
So we have a few questions. So one is the solubility of chitosan. Which solvent you have used?
Glycol acetic acid. Glycol acetic acid.
pH.
Not check, sir.
Then you have performed the drug release, correct?
Yes, sir.
And what pH you have performed?
6.7 something.
6.7 is it okay to support?
No, sir.
But release of fluoride. We don't need to go for the serum. So please check it.
Okay,
And also the XRD, go for the XRD. You got the XRD, what information you want to take to us from this XRD?
Sir, controls compared to chitosan nanoparticles is highest value, sir. It's encapsulate coumarin.
From this peak, we can conclude that it is a crystal or amorphous. So it is a broad band, it is amorphous. Sharp band is crystal. That information only we can take. So okay. Sorry, thank you.
Thank you, sir.
Check the anti-inflammatory trend, right? So, sir, study the anti-inflammatory study. So showing it's your chitosan nanocomposite is anti-inflammatory.
Yes.
So how you going to say it is anti-inflammatory? First, how you induce inflammation in those fibroblasts? You have to induce inflammation, right? The fibroblasts doesn't show dislike any inflammation. You need to induce inflammation, and also you haven't shown any inflammatory markers. You didn't check cytokine level or any inflammatory basic inflammatory markers. And how can you claim this as anti-inflammatory activity? Just showing the viability or just wound closure is not proof that it has anti-inflammatory. You need to show the markers. Then only you can be able to claim that it has anti-inflammatory, right? So go and check what are the inflammatory markers that you need to study first to claim your composite is anti-inflammatory. You just choose the cell line, check their viability, and then you will show one more thing, the wound closure, right? It's just a cell viability, right? It shows cell viability. Right? But doesn't, as a proof that it is anti-inflammatory. Okay?
Thanks, everybody. The next person, Anish Ruben. After Anish Ruben, Mohan Raj, BCRI. Read it. Okay, get ready.
Hello. Hello everyone. I am Anish Rubanes from Biomaterials University. I am here to present my preliminary work on a species I found which was interesting. This is the evaluation of phytochemical and pharmacological properties of Phyllanthus candoleanus. So the systematic question of Phyllanthus, it comes under family Phyllantheaceae, and it is a member of the very famous Phyllanthus species. Like we all know Phyllanthus emblica, which is gooseberry. It has a lot of medicinal properties, and so there is a reason for which I've chosen this plant, because the genus Phyllanthus has been used in medicinally in ethnopharmacological property. It has a lot of ethnopharmacological properties, and it's used in ethnomedicine throughout the world. Like a lot of tribal people, and it's very underutilized, but it's also very prevalent among minority populations, I can say. Yeah, so in a lot of countries, these species are used, and yeah, there has also been a lot of compounds isolated from other species of the Phyllanthus genus. So my objective for the study was to conduct preliminary work to collect, authenticate, and extract Phyllanthus candoleanus bark, and to perform in vitro antioxidant studies, and to quantify and qualify the phytochemical profile of this plant, and to assess the anti-inflammatory activity using in vitro assays, and also to do anti-diabetic assays. So this was my experimental design. Yeah, like from pretty much what I've I've set as objectives, I put in as experimental design, and I'll jump straight to results. So the quantitative screening showed good amount of phenolics in both fresh and dry bark. So for collection, the plant was collected from Nilgiris in around November, and fresh bark was also extracted, and bark was also dried, and extraction was also performed in dry bark, and the phenolic contents and amino acid tannin contents were found to be significant. And yeah, I've also quantified the carbohydrates. So the carbohydrate content was found to be more significant in the dry bark, and proteins and amino acids were also quantified, and it was found that in methanol extracts of the dry bark, proteins were predominant, and in the aqueous extract of dry bark, amino acids were predominant. Yeah, phenols, tannins, and flavonoids were also quantified, and similar results were found. Like methanol extract, gave us a lot more total phenolic content, and meanwhile, tannin extract, the fresh bark was found to have more tannins. And then in vitro antioxidant analysis was also performed. So in vitro antioxidant, like the three assays I did 3 assays: phosphomolybdenum assay, FRAP, and then metal chelating activity of PTA, and in which it was found that in phosphomolybdenum assay, fresh bark extracts of ethyl acetate performed well. Meanwhile, in FRAP, dry methanolic extracts performed well. Meanwhile, in metal chelating activity, again fresh ethyl acetate extracts did better. And then in vitro anti-inflammatory assays was performed. In vitro anti-inflammatory assay, I did membrane stabilization study using goat blood, and then in this also methanolic extracts of fresh barks performed really well. To say it performed really better than the control we used, which you can see here. And then in vitro anti-diabetic studies was also performed, because since it gave really good results in anti-inflammatory activity, I wanted to make sure whether it had any anti-diabetic activity, which was found to be a bit disappointing. And yeah, conclusion. So Phyllanthus candoleanus is a rather underutilized plant in a genus which has been explored a lot. So there has been no proper quantification of its phytochemical profile or anything. So I choose this plant to begin work with, and it proved worthwhile till now. And then that's it. For future plants, I am also planning on developing cell line work on the anti-inflammatory activity, and I'm also planning on characterizing the compounds present in the extracts and isolation. Thank you. That's it.
So we want to perform anti-inflammatory by how?
Further work.
Yes.
So anti-inflammatory,
Since
protein.
you have shown, go back to your show antidiabetic slides.
Yes.
You have shown alpha glycosidase activity is not effective.
Yeah, yes, sir. So compared to control, it's pretty.
Then why you want to study antidiabetic?
Not anti-diabetic, sir. Anti-inflammatory showed very significant results. So I'm planning on doing protein expression studies in anti-inflammatory cells.
So what models? Inflammation is connected to many diseases.
Yes.
Cardiac, even in diabetic inflammation or cancer. Involved. So what model you want to?
In cancer cell lines, sir,
you want to study in cancer.
Yes, sir.
So cancer, as such, the cell line doesn't expose inflammation. Yes. So you have to. I
have to induce inflammation first, and then I will study it. Also, like since the anti-inflammatory activity is really good, it can also have an effect in the cancer pathways. So I'm also planning on.
Inflammatory. See, with this result, don't claim that it has
Okay.
activities. Means you have to go through even.
No, sir. This is only preliminary studies.
Some blood analysis doesn't do anything. Just it will give you a clue. That's it.
Okay. Okay.
Based on this result, you don't argue that. Still, come to a conclusion that it has anti-inflammatory activities. So based on this, so go details.
Okay, sir. I am planning on working in more in detail.
Definitely, seems like it has a lot of active ingredients out there.
Yes, sir.
There's a lot of
But I am also planning on first characterizing it so that I'll get an idea on the type of.
Definitely, it does antioxidant for sure. But you need to approach a right approach to do that.
Okay.
Thank you so much.
Thank you.
Yeah, you go. Thanks for your presentation. Your PPT is good. And I have to post it. So just mention. Actually, this is the because in your data only table and bar diagrams only you have. We leave you have this one. But whenever you go and present in front of the faculty members, at least you should be the photographic witness. So that is the good thing. So only bar diagram, only tables, we believe you, but it is not how to say, the more confident work or something if you want to convey, that witness is needed. Right? Next one, hexane you use for the antioxidant studies. I'm wondering the hexane is not visible with me. Of course, materials. So now you have performed with hexane. Hexane is an organic solvent. When you are mixing the fluidics and other things, 2 layers only we can separate. So please go in the. Thank you. Now I request Mohan Raj, BCRA, Terminal. So please go ahead.
Pleasant greeting, everyone. Myself, Mohan Raj, an UG scholar of Tirunelveli Veterinary College. I am going to discuss on the topic of in vitro anthelmintic assessment of Rhinospora cardifolia nanoparticle against strong-eyed nematodes. Before going to the topic, I am going to give a brief note on our project. That is, goat and sheep are called postman cow because rearing them gives a substantial income and employment to the rural people. About 37% of the world sheep population and 56% of world goat population are reared in Asia. In the recent livestock census, goat alone contributes 27.8% of the total livestock census. Helminth are the common parasite that is affecting goats and sheep. These parasites live on the host for nutrition and protection and causing some illness to the host. The helminths are generally classified into nematodes, trematodes, and cystodes. Among nematodes, our area of concern is strong-eyed. Why I have chosen strong-eyed is it is the most prevalent helminth in the animal community, and it is more prevalent at rainy season and post-rainy seasons. And one female larvae may lay up to 10,000 eggs per day, and it sucks. And one adult parasite sucks up to 50 microliter of blood per day. That is, one sheep with 100 adult parasite would lose up to 50 ml of blood per day. These are the symptoms caused by helminth, that is submandibular edema due to hypoproteinemia and pale mucous membrane due to severe anemia, and death is noticed. The effective control of the internal parasite is very difficult for a veterinarian. To overcome this, and the detrimental effect of the strong-eyed are low fiber production, impaired fertility, decreased milk production, weight loss, and retarded growth, which is causing severe economic loss for the farmers and the country. To overcome this, we are using broad spectrum anthelmintics for a long period of time. But nowadays, resistance to this anthelmintics has been noted around the world. And these are the anthelmintics, and resistance has been noticed all around the world. A fact that India spends almost $103 million per year to control this strong-eyed infections. So that because of resistance, the interest on herbal products has been increasing significantly, and it is used to overcome the resistance. But before using the herbal products, we have the doubts regarding its efficacy, penetration, toxicity effect, and bioavailability. So that we come up with the boom, that is nano drug delivery system. The plant contain active biochemical components that is having many beneficial effects, but it is masked by poor bioavailability and poor absorption. To overcome this, we have used nanoparticles, that is nano-loaded herbal product. The nano-loaded herbal product have advantages than herbal products that are facilitate crossing biological barriers, increase bioavailability, increase reuptake, and extend drug life. So to utilize this advantage, we take our weapon, that is Tynospora cordifolia, locally called as sindil, and it is a climbing shrub. It is indigenous to India and it is used in Indian Ayurveda for a long period of time. And coming to our project, the plant's leaves and stems are collected in Erode and identified by the Siddha College in Tirunelveli. And extract was prepared. The Tynospora cordifolia stem was shade dried for 15 days and it is finely grounded. 25 gram of powder was mixed with 100 ml of distilled water and it is filtered in Wattman filter paper. This is the processing pictures. This is strained, grinding, proper weighing and aqueous extract. And it is filtered in a filter paper and the extract is stored at 4-degree refrigerator for further investigations. Then nanoparticle preparations. We have chosen silver nitrate for our study. 0.1 molarity of silver nitrate was prepared and 10 ml of aqueous extract and 240 ml of silver nitrate was added and kept at room temperature for 15 minutes so that the formation of nanoparticle occurs. The nanoparticle characterization was done by color change, by measuring the particle size, by measuring the zeta potential. According to Henry, the color change from colorless to brownish color indicates the formation of nanoparticle. And determination of particle size was done by zeta size analyzer and its average particle size was found to be 2.8 nanometer. And this is the particle size analysis by dynamic light scattering and zeta potential was measured and it is found to be minus 0.3 MV, which indicates the stability of the nanoparticles. And phytochemicals are screened according to the standard method of trees and events. The phytochemical analysis reveals the presence of saponins, terpenoids, flavonoids, tannin, glycosides, and cardiac glycosides. These are the list we screen for phytochemicals. And these are the pictures of that. And collection of fecal sample. A pooled fecal sample are collected perirectally from sheep and goat of various southern parts of Tamil Nadu. And a weight mount examination was done and the arrow marks are indicating the strong egg. And the egg was separated from the debris from the dung by sedimentation, series of sedimentation tests and flotation technique. This is first sedimentation followed by second sedimentation and flotation technique using saturated salt solution and a serial sedimentation was done. Egg suspension was prepared using centrifuge. Then egg hatch assay was performed to evaluate the ability of plant extract to inhibit embryonation or hatching of nematode egg. A microtator plate was 24 microtator plate was taken and extract of various concentration was added, that is 10 mg, 20 mg, 40 mg and 80 mg. And egg suspension of 100 microliter were added to each well. And distilled water was added to make up 2 ml. And then positive and negative control was taken. For positive control, fenbendazole, that is standard anthelmintic drug, was used at 0.1 microgram per ml. And for negative, we have used distilled water. And we have taken separately herbal extract and nano herbal extract to identify the difference among them. And after 48 hours of incubation at room temperature, Lugol's iodine is added to prevent the embryonation, further embryonation. After adding Lugol's iodine, using inverted microscope, we have counted the unhatched larvae and unhatched eggs. And the percentage of inhibition was calculated using this formula. And these are the control. The first one is positive control, that is using standard anthelmintic drug, and second one is negative control using distilled water. And you can see clearly see the motility of the larva. But in a positive control, there is no larva is present, only egg is present. And coming to the result, the positive control showed 94% of inhibition, and at a high dose, that is 8%, aqueous extract showed 76%, and nano extract showed 90%. Now you can notice the difference between the aqueous extract and nano extract at 8%. The aqueous extract showed 8%, inhibition of 76% at 8%, and nano extract showed 90%, which is highly differentiable. And look at the 8% inhibition at inhibition of 90% in 8% well, and compare the positive control, which is the difference is only 4%. So I conclude that the nano extract is almost equal to the standard anthelmintic drug, and it can be act as a replacer for the anthelmintic drugs. And the images are plotted in a graph, and you can see the positive control and nano extract at 8%. So the conclusion is we have done in vitro study, but in future we have planned to do in vivo anthelmintic activity of this plant. And at in vitro study, I strongly concluded that the Stinospora cardifolia plant has anti-nematodial activity, and the principal constituent for this anthelmintic has not been identified. It will be carried out in further studies. And acknowledgement, thanks to the Dean and Head of the Department of Pharmacology. And this project is funded by TNSCST. And these are my references. Thank you.
If this plant is edible, this goat and sheep, they may eat this plant.
Yes, sir. They will.
They are edible, right? Then naturally they will develop this activity.
No, sir.
Activity, right?
Yes, sir. They will eat, sir. Usually for the farmers are using, sir. But I'm scientifically proving that.
You're proving it scientifically, but people are already using.
They are using, sir. Yes, sir. As an ethno-veterinary, they are using.
Seen, why don't you check with the extract alone for the nano?
Yes, sir. I have checked for herbal extractor.
Extract alone.
Alone. It showed a 76%.
Only I see the nanoparticle.
Sir, 1 minute. Sir, that is aqueous extract. They showed 76% inhibition, sir. But nano showed 90% efficacy, which is almost equal to standard drug.
What nanoparticle you use?
Silver nitrate, sir.
The silver is toxic, right?
Yes, sir.
Definitely that toxicity, that will enhance.
Yes, sir.
Naturally, it has 75%.
But
that is good.
We are using it at 3-month one interval, sir. So,
sir, silver is, I think, it's not still a FDA approved drug, sir. Already that extract is better. Better you look for some biodegradable nanoparticles.
yes, sir. But it's a cost effective and so we
expensive than other biodegradable, sir. Right? Silver is expensive, right? Per gram is 60, 70 rupees around. Look for some biodegradable, that will be much better.
Okay, sir.
Okay, thank you.
Thank you.
Anyway, you are looking good. So, well, do better. Good.
Thank you, sir.
Don't mistake me to say a few things. As a chemist, nanotechnology, I'm working more than 20 years in this area. But even nanomaterials, I guess it is not completely formed. The reason is you have given 2.8 nanometer size. There is no way to produce the nanoparticle that size. If the particle size is less than 6 nanometer, it's called cluster. But the color changes will not be occurred. But your case is occurred. So I guess your zeta potential you diluted lot. That is the reason. And also you have given the zeta potential value is minus 0.3. According to the literature review, if the zeta potential is plus or minus 25 millivolts, higher than 25 millivolts, then only the particle is stable. So please go very in-depthly in this study. And also the same image and your zeta potential value both are somewhat contrast. Okay. Study more, and as I agree with our Professor Ashok Kumar sir. So there is a problem in the formation of nanoparticles. Please refer with the nanotechnologies. All of this. Thank you. The next person is Kruti. Yes, sir. Okay. Then Archana Rajavel, research scholar. Yes, sir. Yes, please come. After Archana Rajavel, Rishika Nair, yes, be ready. Vikas, okay. Prakash Krishna, Maria.
Happy afternoon to Nanotechnology Center. Hi, hello, I am Archana Rajavel from SRM, research scholar. I am going to present the topic FTIR and MALDI-TOF detection of extracellular vesicles, a rapid detection tools for Duchenne muscular dystrophy. Before we go to our topic, we need to know about the two keywords here, Duchenne muscular dystrophy and extracellular vesicles. Duchenne muscular dystrophy is a rare and devastating muscular dystrophy. It is caused by out-of-frame mutations in the DMD gene. It is the second largest gene in our body. Dystrophy that maintains the structural integrity and myofiber function. 1 in 3,500 live birth males are affected worldwide. But for our India, we don't know about the prevalence rate. The onset of age is 4 to 6 years, and they lost their ambulation. They can't able to walk at the age of 13 to 15 years due to the muscle degeneration and early death complications due to the cardio and respiratory problems. And that extracellular vesicles, he is the hero of our project. Why we select him? Because based on the some important biological features of him, and the nanolipid layers are 20 to 200 nanometer size. It is easily accessible and permeable, and it evoke a low or no immune response. It is an intercellular communicator. It carries the molecule content of the parent cell to the neighbor cell to communicate with the neighbor cell. It crosses the blood-brain barrier membrane and delivers the drug to the target. It is used as a potential surface protein, and receptors are used as a biomarkers for diagnostic and monitoring the disease progression. Here is my objectives, 3 main objectives. We study the protein subset of molecular content of extracellular vesicles from the samples of DMD patient, and we use FTIR and MALDI spectra can discriminate the normal healthy individuals and DMD patient to identify the potential biomarkers for Duchenne muscular dystrophy by MALDI-TOF MS. The subject details, we took a total number of samples 20, cases 10. Among them, the ambulant is 5 and non-ambulant is 5. And the control group is 5. They are age-matched control. Here is our methodology. Here we use the kit-based method. We took the blood samples from the patient and the process to get the clarified plasma, and we isolate that exosomes by adding the precipitation method, and we characterize it in TEM, DLS, and FTIR. Then we study the protein biomarkers in MALDI-TOF MS by mix it with matrix under certain parameters, then analyze the protein patterns compared to the normal and the DMD patient. Here is the result of our biophysical characterization of plasma LEVs. Based on the precipitation method, here we use a less volume of a sample. The total time to process the sample is 2 hours. The purity is moderate compared to that gold standard method ultracentrifugation. The yield is high. In zeta potential analysis, we got a nice result, like the range of -40 to +40, and the particle diameter is ranges from 100 to 300. Here in 10 micrographs in 100 nanometer size, 100 nanometer scale, we got that image of intact exosomes, and we use that images software to count the number of particles present in the particular amount of volume of sample. Here is the FTIR characterization of plasma-derived extracellular vesicles. Mainly, we focus on the fingerprint regions. Here is the 3 different categories. One is normal, an ambulant patient, non-ambulant patient. Then we analyze it in the origin software to plot the PCA analysis. Then we can discriminate the normal and the DMD patients based on the spectra derived from the FTIR. In multi-MS spectra, we can distinguish that normal and DMD patient extracellular vesicles based on the fingerprint, study the fingerprint regions of the exosomes. And we got 20 different peaks, and we average it in average and comparator to DMD and normal. Here, some of the protein based on the mass is differ in that both normal and DMD. And it's already reported in other studies that involved in cancer and cell line studies. So we, for the reference for extracellular vesicles, we got it from there. And plasma proteome database also reported the extracellular vesicle proteins. That is act as a potential biomarker for inflammation and muscle related problems. And here we got a C4A complement apolipoprotein C3, C3 complement component 3, and apolipoprotein C2. And we conclude, we propose it's an easy and alternative approach for solid biopsy, and it is a FTIR candidates to be an alternative and complement method to discriminate the normal and the DMD population. And the multi-MS is a potential technique to study the fingerprint analysis and analysis of exosomes that are from both the normal and DMD patient. And the future direction is the mass spectral fingerprint database and validate the potential of biomarkers among a large DMD cohort and monitor the biomarkers in different stages of DMD patient and to propose a new theranostic approaches for DMD.
What is the significance of your study, ma'am?
So it is an alternative method for solid biopsy. Usually for DMD, they took the muscle from the patient and do immunohistochemical study. So it is an alternative method, liquid biopsy. For that, we need a very small amount and it's a less invasive.
Yeah, how much percentage you would say like this extravascular, I mean, this extracellular vesicles are whatever it is, how many percentage of CS this person is affected by this muscular dystrophy?
One is that
how much percentage is the surety that okay, this person have muscular dystrophy?
based on FTR model.
Based on your monitor from this.
Yes, sir. Actually,
Here it's there, it is just random. Maybe a muscular related diseases. It's not specific that this person is having a muscular dystrophy.
Because it has contained 5 different stages: pre-symptomatic, early ambulatory, and late non-ambulatory. So we segregate based on that, and we collect more samples. Then only we short tell that it is a biomarkers for Duchenne muscular dystrophy.
Okay, ma'am. Because since this MDS gene, I mean, mutation is causes this disease. So directly, if you do a PCR, we can able to identify.
But the cost is high compared to.
PCR is cheaper than doing molecular.
No, sir, it's an alternative approach.
That is, I accept that point. It's an alternative approach. Yes.
And we study
the. You want the confirmation, right? Because go to a doctor, right? See what you say. If he give a medicine, you say 100% yes. I need to be cured, right?
Yes, sir. But it is very heterogeneous. By the mutation.
You go try today. Whether your fever is reduced, okay, fine. Otherwise, you come tomorrow. No doctor is says that, right? But you do more studies. Make sure. Yeah, this approach is good. I'm not saying this approach is good, but make sure it need to be 100% efficient. Yes, good luck.
Thank you, sir.
Yeah, question.
Yes, sir.
Your FTA data, can you go now? So all are matched together. We cannot able to distinguish which peak and also see the lot of noises are there.
Which
separate the things one by one. I hope you have seen many data available in that infection.
The stack kind of thing.
You are expert to use origin. Why don't you squeeze this one? Otherwise, a lot of noises and the peak prediction is not good.
Okay.
Go for the slide number 14. Yeah, what is in y-axis? There is no detail. So this is MALDI data, but you haven't shown any MALDI data. Only the diagrams.
I have it in my bag.
Okay, no problem. So you have to give whenever you are giving the presentation, this detail should be.
Yes.
Good luck.
Thank you.
The next question, because No, sorry, Rishika Nayak. Yeah, research scholar VIT. After that, Vikas.
Good afternoon, everyone. I am Rishika Nayak, pursuing my PhD from the Department of Electrical Engineering, Vellore Institute of Technology. And my PhD work focuses on heterostructured graphitic carbon nitride by TiO2 nanocomposite to evaluate the photocatalytic CO2 reduction competence under visible light. So introduction, by now everybody might be aware of what a nanoparticle or what a nanomaterial is, and there are various applications. So my application of nanoscience and nanotechnology have attracted the colossal interest due to its high catalytic activity and selectivity in the field of energy and environment. And as we can see that our climatic conditions are deteriorating, it is very important that we find a technology or a method to bring the CO2 concentration in the country or in the world under control. And for this, we can see the global energy consumption data. And if this trend continues, IPCC 6 assessment have reported that by 2100 and by 2040, the temperature may rise by 1.5 degrees Celsius. And in order to bring this under control, in COP21, a collective effort by all the countries have been taken that we will bring the global warming below 2 degrees Celsius. So in India, the Prime Minister has set his country a target of net zero greenhouse emissions by 2070. And in order to achieve this, the first thing that we have to do is to control the CO2 emissions and to sequester carbon dioxide, and we can convert into various fuels. So different types of CO2 reduction, it can be thermochemical, it can be using an electrochemical, and can be using biological. But my area focuses on photocatalytic CO2 reduction. So simply photocatalytic CO2 reduction is similar to that of photosynthesis in plants. Like how plants take up carbon dioxide and water and sunlight and convert it into glucose. Similarly, a photocatalyst is a semiconductor device that absorbs sunlight and converts into value-added fuels like it can be methane, methanol, ethanol, etc. So it usually consists of 5 steps. That is, the first step is light absorption. So when the light is absorbed, there is charge separation. That is, electrons get concentrated in the conduction band and the holes get concentrated in the valence band. So once this electron separation is achieved, the third step is CO2 absorptions. And this, once the CO2 is absorbed, the fourth step is surface redox reaction. That is, the electrons contribute to CO2 reduction to different fuels, and the holes contribute to water oxidation. And once this is achieved, the fifth step is product desorption. That is, the methanol or the produced fuel has to be taken out from this solution in order to avoid catalyst poisoning. So my objective is to synthesize graphitic carbon nitride and TiO2, and then to create a heterojunction between graphitic carbon nitride and TiO2 and test its efficiency in photocatalytic CO2 reduction, and to understand what is the mechanism involved in this catalyst. So the methodology is, the first I'll be preparing the TiO2 and graphitic C3N4 separately, and I'll be creating a heterojunction in the composition of 1:2, 1:1 and 2:1 ratio. And then I'll be doing various characterization techniques to prove that the nanocomposite has been successfully achieved, and then utilizing this nanocomposite for CO2 reduction. So nanophotoCatalyst synthesis, first I have used titanium tetraisopropoxide as precursor and ethanol, and in solution B will be ethanol, water and acetic acid. So this is the method I have used for synthesizing. Then the characterization results. So first is XRD. So the XRD results will help in understanding whether whether the catalyst is highly crystalline or not. And the second is if the composite has been formed. So we can see in the TiO2, the different peaks, that is at 25.4, etc. All these peaks, according it matches with the JCPDS card number, and it proves that the TiO2 in its anatase form has been formed. And this peak in graphitic confirms that the graphitic carbon nitride is also formed. And in the composite, we can see in the 2:1 composite, there is TiO2 peaks as well as graphitic carbon nitride peaks. So this suggests that the composite has also been formed successfully. We can see that the crystalline size also, and we can see a descendant tendency in the crystalline size. And we can see in 2:1, the crystalline size is very less, from which we can conclude that the TiO2 has successfully nucleated and grown on the surface of graphitic carbon nitride. The second is FTR analysis. So this stretching band at 500 to 900 centimeter in the first one, that is the red color line, shows that the TiO2 has been formed successfully. And further in the composites, the steep band and all these other peaks at 1,248, 1,326, etc. shows that in the composite also TiO2 and graphitic carbon nitride has been formed successfully. Next is the UV analysis. From the UV analysis, we can see that the catalyst is active in 200 to 450 nanometer region. And using this absorbance data, I have calculated the band gap using TOC analysis. So from the band gap can be seen respectively, and from using molecular electronegativity equations, I have calculated the conduction band and its valence band potentials also, and even found out what wavelength the catalyst is highly active. And from the FP7 EDX analysis, at 1 micrometer range, there is agglomerated TiO2, and at 500 nanometer range, we can see the spherical structures of TiO2, which confirms that TiO2 has been formed. And in the EDX analysis, the peaks confirm that the TiO2 has been successfully formed and there is no impurities present. Next is the graphitic carbon nitride. At 500 nanometer, we can see that the flakes-like structure or layers-like structure, and it has been aligned very beautifully, and this signifies that the graphitic carbon nitride has also been formed. And similarly, the EDX proves that there is no other impurities other than graphitic and carbon nitride. The third one is the composite one, that is in the 2:1 ratio, and this also proves this roughness in the graphitic carbon nitride layer proves that TiO2 has been successfully formed in the layer, and even the EDX proves that TiO2 and graphitic carbon nitride has been formed. So this is how the CO2 reduction setup was. So in this I have added the catalyst and NaOH, and it is kept in the dark medium. And after every 1 hour, I have sampled out 5 ml, and using the CO2 analyzer, I have calculated the CO2 concentration in the medium. So this is the CO2 reduction analysis. From this normalized graph, we can see that the TiO2 has shown the least efficiency in the visible light region when compared to graphitic carbon nitride. And this is because the TiO2 is very less active in the visible light region when compared to graphitic carbon nitride. And other possibilities are because of the formation of electron hole pairs, that is formation of free combination. When compared to the composite, we can see that 2:1 shows the highest CO2 reduction efficiency and demonstrate that there is the vital junction between GC3N4 and TiO2, leading to better stabilization of the catalyst and improvement in the separation of photogenerated electron and holes that further activates the reduction in the CO2 in composite framework. So there are two proposed mechanisms based on the literature. One is type 2 heterojunction and the second one is ZSK mechanism. So both the mechanisms, the first step is as soon as the light falls in the semiconductor, there is electron and hole pairs creation. So once the electron and holes pair are created, the electrons will move from the valence band to the conduction band in TiO2, and similarly in GC3N4 also. But what happens in type 2 heterojunction is the electrons from the conduction band of graphitic C3N4, since it is more negative than TiO2, it will move towards the electrons in the TiO2, and the holes from the valence band of TiO2 will move to the valence band of graphitic carbon nitride, and successfully TiO2 to CO2 will be reduced to different fuels like methanol and methane, and in the holes there will be water oxidation. Whereas in ZSK mechanism, what happens is, since the applied electric field is from the graphitic C3N4 to TiO2, there won't be any other electron hole pair generation or any other electron hole pair separation. but the only electrons will move from the conduction band of TiO2 to the valence band of graphitic C3N4, and from the valence band of graphitic C3N4, the electrons will move, and the CO2 reduction will happen in the graphitic C3N4, and water oxidation will happen in TiO2. So this is the mechanism that is being provided theoretically in the literature survey. So hereby I conclude that from the characterization results, it is proved that the heterojunction has been successfully formed, and even the CO2 reduction efficiency for the composite is much higher when compared to individual compounds. That is because there is a good electron hole pair separation, the crystallite size, and its high ability and its high efficiency to absorb light in the visible light region. So this is the main reason the main reason behind the composite catalyst to show a better CO2 reduction. So my further work will be on to find out what are the fuels produced using GCMS. I'll have to do what fuel production is there. That's my further work, and I would like to thank Vellore Institute of Technology and my center, that is CO2 Center and Green Technology Center, School of Electrical Engineering, and my guide Dr. Velbidiji for their immense support towards my research work. Thank you.
So I have two questions. So you have taken 1:1, 1:2, and 2:1 ratio.
Yes, sir.
But all the cases you have been observed unattest phase.
Yes, sir. All the cases there is unattest phase.
What is the reason?
So because initially I have used the same TiO2 for composite synthesis also, sir. Initially,
you have prepared TiO2 alone and the C3N4 alone.
Yes, sir.
You mix together.
Yes, sir. I did multipestle only, sir. I didn't go for homogeneous synthesis. I have to do homogeneous. I just went grinded it in multipestle and then I calcinated it.
But in SEM, it is well known that graphitic C3N4 will give the sheet-like structure and a hollow structure. But in your case, we haven't seen anything.
So this is flakes-like structure, sir. This graphitic C3N4, it can be either sheets or it can be hollow, as you said, but it can be even flakes. So this flakes-like structure is also graphitic C3N4, sir.
But you have to do the further confirmation by XPS and other things, right?
XPS, okay, sir, that can be done, but even EDS analysis, sir.
EDS is not a correct confirmation. Even your SEM, the base specimen itself can give the compounds.
Okay.
Yeah, the XPS is the correct correction.
Okay.
And one more thing, the band gap is higher in the 2:1 ratio. What is the reason?
So band gap is lower in 2:1 ratio, sir, when compared to all the compounds. Lower the band gap, the ability.
2:1.
Yes, sir.
Yes. So what's the reason?
So that is because the graphitic carbon nitride composition is higher. The graphitic carbon nitride band gap is 2.78. So and TiO2 is 3.11. So in 2:1 ratio, graphitic carbon nitride is 2 part and TiO2 is only 1. So that's why its composition is band gap is higher.
But based on your observation, you are saying that 2:1 is the leading one, correct?
Yes, sir.
So the low band gap, high energy is given.
Yes, sir. That is because graphitic carbon nitride and TiO2 compared to crystallinity, TiO2 is highly crystalline and graphitic C3N4 is less crystalline. So when we do the composite, this heterojunction will help in giving better CO2 reduction efficiency.
Okay, the next one, go for the experimental section. The CO2 gas analysis.
This one?
So you purging
Yes,
CO2 gas.
Yes.
But it is not a closed one. When you are purging, see the setup release the SMR. There is a chance to come again the gases.
So actually, this is not online CO2. First, I will purge it once I completely seal it with paraffin wax, and then I'll purge CO2 in closed condition for 15-20 minutes. After that, I'll take the setup to the CO2 reduction with the cork. There'll be a cork on top, sir, and I'll be taking out the sample.
When you are purging, it should be mixed together. With the high pressure only it can mix together. Currently, it is a gas, and it should become a liquid. It will not come without giving the pressure. So did you perform that thing?
So pressure I haven't checked, but the another thing is I have added NaOH. So this NaOH will actually help in dissolving CO2 in the water medium easily. That's why.
Okay, thank you so much. The next person.
Thank you.
Vikas.
Good afternoon, everyone.
Prakash Krishna is there? Genevieve Maria. Then from Chettinadu, Pallavi. Then Kauda. Okay.
Good afternoon, everyone. I am Vikash, undergraduate student from Madras Veterinary College. Today I am going to discuss about in silico analysis of broad spectrum gastrointestinal selective antibiotic macrocyclic lactome against canine parvovirus. Canine parvovirus is a highly contagious and relatively common disease in young and unvaccinated dogs. There is no specific treatment for canine parvovirus, only symptomatic treatment is available. Canine parvovirus is a non-enveloped single-stranded DNA virus which enters the host cell via endocytosis. This is the mechanism we are going to target. There are various subtypes in canine parvovirus. It is actually evolved from feline virus. In 1978, it is emerged from feline parvovirus, and now it is a canine parvovirus. Presently, canine parvovirus 2C is available. This is the life cycle of canine parvovirus. Infected dog will excrete the virus in the feces. The healthy dog will ingest the virus through oronasal route. It will multiply in the dog's intestine and it will establish the infection. With successful treatment, animal recovers. But if there is no supportive treatment, it is a very prolonged period. So we need a more specific treatment. Clinical signs: there will be lethargy and there will be vomiting, persistent thing, and hemorrhage will be there. Diagnosis: in field level, the only diagnosis available for canine parvovirus is based upon symptoms, which is very characteristic foul-smelling diarrhea. Therapy: you can notice there is no specific treatment for this virus. Only symptomatic treatment is provided, restoration of fluid and preventing secondary bacterial infection. So we came with the concept of in silico analysis for a specific treatment. The receptor is the protein which is available both in host and also in drug. Ligand preparation: we took different macrocyclic compounds from PubChem. This is the 2D images of various structures of macrocyclic lactones. Protein preparation: we took transferrin receptor protein and antibody fragment D and capsid structural protein of virus from protein data bank. This is the transferrin receptor protein which is present in the host, which helps in establishing the infection. This is the antibody which is produced by the host to neutralize the virus. This is the capsid structural protein which is present in the virus, which is helpful in establishing the infection. The software we use is Pyrex Python, which is used to run the program and also visualize the results. Crescent flare, which is used to find the active sites of interaction between ligand and the receptor. Osiris data variable is used to find the flexibility, hydrogen receptors, hydrogen donors, polar surface area, triglycerides, tumorogenic, mutagenic characters. These were the results we obtained. You can see among various components, milbemycin is having a very good binding energy and is also less mutagenic, tumorogenic, and also less irritant. These are the visualization from our softwares. So among the various macrocyclic lactones, we chose milbemycin because it has a good binding affinity with transferrin receptors and antibody fragment and also capsid structural protein. It is also less tumorogenic property. So further in vivo studies should be carried out to prove its effectiveness. Why we chose in silico? Because it is less cost effective. It is a less cost effective way, and it also helps to overcome the barriers of clinical testing. It gives advantage because there were so many drugs, but we chose one drug. If we go for the many drugs in an animal, it will be very menace. These are the references.
Okay, Vikash, thank you so much for your presentation. So I have two questions. So you just shown some diagrams, right? This one you just taken from internet or you draw by yourself?
No, sir, from the software.
From the software.
Yes, sir.
So go for the structure of milbemycin. So you have mentioned here it has the good affinity with the receptor and all things.
Yes,
here many functional groups is there. In which group have the high affinity with the receptors? C here ON and COH like this.
So the NO.
Which one?
NO. NO, yes.
Okay, thank you so much. Then next person, Prakash Krishna from Madras Veterinary College. After Prakash Krishna, Genevieve Maria. No, you just go for your results directly and shortly click your introduction.
Hi, good morning to everyone. I am Prakash Krishna from 4th professional year, Madras Veterinary College, Tanvas. And the topic that I've come up with for today's presentation is an innovative in silico analysis of antifungal products, namely triazole derivatives against canine malassezia patchy dermatitis. I'll just go a brief of the introduction. Canine malassezia patchy dermatitis was first isolated in early 90s by Fred Wellman. These are lipophilic yeast. Currently, there are 16 species of under the genus Malassezia. Malassezia are commensal organism in dogs and cats. The overgrowth is almost and always associated with an underlying primary cause. These were the clinical signs of Malassezia, which include generalized alopecia and intense erythema. And we could observe lichenification and hyperpigmentation on the entire ventral regions. These Malassezia were opportunistic pathogens which were also present in humans, where causes hyperpigmentation and hyperpigmentation in humans, including dermatitis, dandruff, folliculitis. I have added this article which states that Malassezia patchy dermatitis was reported as an zoonotic organism. For the first time, we are using triazole derivatives, which are antifungal medication often used against Candida species in humans, against cytochrome P450 and linosterol 14-alpha demethylase receptor. In order to know about this receptor, I would like to explain the mechanism of action in a gist. This is the fungal cell, and the yellow color molecules are the ergosterol, which are responsible for maintaining the stability of the cell wall. The blue color compounds are linosterol. Because of the 14-demethylase and cytochrome P450, linosterol is converted to ergosterol, thus it's responsible for the stability of the cell. Because of triazole compound gets bind to this 14-alpha demethylase and alters the structural activity specific of a 14-demethylase receptor. Thus, the ergosterol is not formed, which results in destability of the fungal cell. Triazole derivatives have broad variety of actions like antimicrobial, antiviral, antitubercular, and etc. Antifungal properties have been discovered recently. The triazole derivatives include voriconazole, fluconazole, posaconazole, isaconazole, and itraconazole. We obtained the protein from Uniport database, and the triazole derivatives were obtained from PubChem and were converted to PDBQT format. We used Pyrex Python prescription software and Crescent Flare and Oris Data Warrior for carrying out the further drug parameters. I have tabulated the properties of triazole derivatives with compounds of cytochrome P450 and linosterol 14-alpha demethylase receptors. The first figure exhibits the binding of fluconazole with cytochrome P450 receptor with the binding affinity of minus 8.2 kilocalorie per mole. The active site of binding were found to be at 3 positions, namely isoleucine 376, triosine 515, and methionine 518 receptors. And I have added isavuconazole with cytochrome P450 and itraconazole with cytochrome P450 with a binding affinity of minus 14.5 kilocalorie per mole, and posaconazole with cytochrome P450 with a binding affinity of minus 14 kilocalorie per mole. And this is the second receptor protein, that is linosterol 14-alpha demethylase with the above triazole derivatives. All these compounds obeyed Lipinski's rule of 5. Among the triazole derivatives, tetraconazole was found to have higher binding affinity with both cytochrome P450 and linosterol 14-alpha demethylase, and followed by posaconazole. Further, to demonstrate its efficacy, in vivo tests should be performed. Thank you.
Thanks, Professor Krishna. The next person, Genevieve Maria. Remaining 2 persons are there. Prakia Pallavi, Chettinadu, Kaudam,
Genevieve Maria Mirman. Photocatalytic activity.
Did you come copy?
Yes. Today morning.
When I
Yes,
can't.
yes. Not this one.
Can you change it?
Good afternoon, everyone. I'm Genevieve Maria Mirgam, research scholar from STNB Vaishnav College. My topic is photocatalytic. My topic is photocatalytic dye degradation and bacteriostatic efficacy of microsynthesized copper nanoparticles by new isolate Lentinus scorosilus from dead trunk of Nerium odorum. This is the image of Lentinus scorosilus mushroom. Introduction. In recent years, nanotechnology has played a significant role drug delivery, biotherapeutics, pharmaceutical and biotechnology, etc. The advantages of mushroom-mediated copper nanoparticles are eco-friendly, safe to handle, and having diversity of secondary metabolites, including terpenoids, polyketides, steroids, and phenolic compounds, which acts as a reducing and capping agent for synthesis of copper nanoparticles. Currently, copper nanoparticles have been attaining great attention due to their catalytic, optical, and electrical properties. Therefore, copper is mostly preferred for the synthesis process because of low cost and high conductivity. Moreover, copper nanoparticles are widely used as antimicrobial, antioxidant, anti-cancer, anti-inflammatory agents. It is also used as an alternative feed additive in animal feed, especially poultry diet. Lesser concentrations of copper are safe for human cells but effective for microorganisms. The copper nanoparticles act against a wide range of bacteria by disrupting cell wall and distortion of helical DNA structures. Textile dye is a rich source of toxic organic compounds. It causes environmental pollution. The azo dyes like Congo red, brilliant blue, and forest green, a main class of synthetic colorants, are usually used in the textile manufacturing due to their properties such as a large variety of shades, resistance to decoloring, and a lower energy consumption. The textile dye enters into the soil and water bodies, affects the ecosystem, and causes environmental hazards. In this walk,
Madam, you can go directly to your expert setup, madam. These are the things we know.
Okay. So, coming to synthesis of copper nanoparticles. Microsynthesis of copper nanoparticles. Mushroom extract is added with 5 mm stock solution of copper sulfate. The color change of brown color indicates the formation of copper nanoparticles. The antibacterial activity of copper nanoparticles. There are 4 bacterial strains used for my study. The inhibition zone. Against this bacteria,
by copper nanoparticles are studied. This is a graph of zone of inhibition graph against the bacterial strains. Here, two concentrations are studied. One is 25 mol and another one is 50 mol, and it is compared with the standard control ampicillin. The highest inhibition zone found in Enterococcus aerogens in 25 mol when compared with control, and medium inhibition zone found in Streptococcus mutans. And in 50 ml concentration, the highest inhibition zone found in Enterococcus aerogens, and medium inhibition zone found in E. coli. Next, photohydrolytic activity. The first image shows the degradation of Congo red dye, and the second image shows the degradation of fast green, and the third image shows the degradation of copper nanoparticle in brilliant blue. Next, the catalytic degradation of different dyes by copper nanoparticles. The graph shows the highest degradation in Congo red dye, followed by fast green dye and brilliant blue dye. The characterization of copper nanoparticles. The copper nanoparticle synthesis confirmed by a characteristic peak obtained at 319 nanometer in UV visible spectroscopy, and next in FTR spectra of copper nanoparticles. The copper nanoparticles were analyzed through FTR in 400 to 4000 centimeter range for the presence of functional groups surrounding them. FTR spectrum of extracts with copper nanoparticles main peaks at 1398.6 centimeter, 1512.4 centimeter, 2038.6 centimeter, 2348 centimeter, 3220 centimeter, 3856 centimeter are present. Bands present at 1233.6 centimeter, 1398.6 centimeter, and 1512.4 centimeter corresponds to ester linkages, lignin and cellulose, and amide 1 group respectively. Band present at 3680 centimeter and 3856 centimeter corresponds to proteins, carbohydrates, flavonoids and tannins. Presence of these groups suggests that they are acting as capping agent, preventing nanoparticles agglomeration. Next, SEM and EDX analysis. The morphology of the green synthesized copper nanoparticles was examined using SEM. The prepared copper nanoparticles were found to be in irregularly flated, needle-shaped and aggregated. The size of copper nanoparticles from SEM analysis was found to be 1 micrometer, shows the presence of copper nanoparticles. The EDX analysis confirmed the composition and stability of synthesized copper nanoparticles. The purity levels of the particles were examined, which indicated the lentiginous corrosulus mediated copper nanoparticles had 3% copper and some weak signals of carbon, oxygen, silicon, sulfur, calcium and aluminum elements. These weak signals may be due to the X-ray emission from the macromolecules like flavonoids, phenolic compounds, carbohydrates, glycosides, steroids and tannins present in the extracts. Conclusion. In the present study, copper nanoparticles synthesized by a simple and benign method from mushroom extract of lentiginous corrosulus. The characterization.
That's not necessary, ma'am. That's okay. We can understand from the results. So, if you have any questions?
Yes, sir. You just come this side, madam. The next person, Rakia Pallavi, please go here. Madam, your case, whatever the stability of your copper nanoparticles, how long it is stable? No, that is the composition from EDX. Stability means you prefer today, after 1 week it is stable, or any precipitate you observed, or any color change you observed. Can you check that one? Okay, it is very important to check. And based on your UV visible and the EDX data, the composition, I mean 3% only is copper is there. So you have added very least quantity of copper. So you should scale up it, then only your activities will be good. So EDX is very important data, and also if you are seeing UV visible, the intensity observance is less. So it is the composition. Okay, thank you, ma'am. Rekia Pallavi, please start. Rekia Pallavi, research scholar from Settinadu Medical Institution. One more presentation is that will be from Gautam, the last presentation. So please try to conclude.
Good afternoon,
Yes,
everyone. Good afternoon, everyone. I'm Pragya from Chettinad Academy of Research and Education. My topic is nanohybrid from photodynamic therapy and imaging. So all we know about the cancers that refer to the cells that grow out of control and invade the other tissues also. So for the cancers, there are so many treatments are available. Mostly commonly available are chemotherapy, radiation therapy, and surgical removal. Besides this surgery and radiation, chemotherapies are commonly available. So chemotherapy also have this so many side effects, like it damages the healthier cells along with the cancer cells. So with the diagnosis with the existing tool, inhibit the poor detections and limit and fail to detect the smaller tumor. Since the cure rate is not satisfied, early diagnosis and effective treatment need to be developed. So here we come to the photodynamic therapy as an alternative tools. You can see in the figure here, for the photodynamic therapy, we need three things like photosensitizer, oxygen, and a proper wavelength light. So here we can see that in body, we are injecting the photosensitizer by intravenous or topical route. Then it will go to throughout the body. Then we'll give the light to the particular tumor site. In particular wavelength, will give the light to the tumor site. There only it will generate the ROS. Then it will help to kill the cancer cell. So PDT mechanism is when they are giving the light to the photosensitizer, it is excited to the single state, then it's going to the higher state, and after that it's really come back to the relaxations, and mechanism they are giving two type reaction, they are giving two type 1 and type 2 reaction. In type 1 reaction, generate the radicals and radicals ion species, and type 2 reactions, it produce the singlet oxygen. So here with photodynamic also have some advantages and disadvantages also. Like with the great progress in the PDT, there is a clinical application that's still hampered due to the limitations regarding the photosensitizer as pore solubility, water solubility, toxicity, and specificity, tumor specificity, tissue specificity. So various nanomaterials have been designed to overcome the molecular drawbacks of the conventional PAs. So here, nanohybrid materials, for their enormous advantages over the individual components, have shown the theranostic approaches. So what is a nanohybrid? Nanohybrid are the combination of two different materials, organic and inorganic. It has the applications in biology, medicines, energies, and other fields also. So the class of nanomaterials retain the advantage of both organic and inorganic compounds and provide the ability to tune the properties of the nanohybrid through the combination of the functional components. So these are the type of nanohybrids. It can be organic hybrid, inorganic and inorganic hybrid. Inorganic and organic hybrid is clay-based nanohybrid, carbon-based nanohybrid, metal and metal-based nanohybrid, and organic hybrid is lipid polymer nanohybrid. So these are some applications of the nanohybrid where we can use in photothermal therapy, photodynamic therapy, and both photothermal and photodynamic therapy together. In imaging also we can have the applications of visible light, NIR, ultrasound, MRI, and CT. You can use in the drug delivery system also. So these are some compositions of the nanohybrid we are using, like organic compounds like dendrimer, liposome, and inorganic compound like mesoporous silica, fullerene, and organic nanoparticle, metal oxide nanoparticles. So with this, we can conclude that due to the limitations of the conventional photosynthesizer, the development of the nanotechnology is making a breakout in the field of photodynamic cancer therapy, in which hybrid nanoparticle contribute a lot by the integrating the advantages of the both organic and inorganic compounds. Some of the nanohybrids can be modulated to the tumor environment to overcome the.
Thank you, Prekia. So it is visible on the conclusion. Anything acknowledgement if you want to give, you can give.
So these are my reference and acknowledge to my guide and my colleagues and Chetnad Academy of Research and Applications.
Okay. Thank you. The last person of this session.
One suggestion, when you come for this kind of international conferences, present some of your results, right? Work and all is okay, fine. Fine. Present some research work that will help, right? Fine. In future.
The research data is provided that we are the editor for many journals. We are giving the comments as the editor. It will be useful for it. That is why we are doing. Okay. Thank you so much. Yeah. Yes. You done well. Fine. Last person of this today's session, Mr. Kaudam from Chettinad Medical University.
Good afternoon, one and all present over here. And my topic is ultrasound response to drug delivery. As we all know that we are, as we know, like every drug we needs to treat cancer having a specific drawbacks, and most of the therapies what we have in the present trend is like having many drawbacks in drug delivery systems, and most of the nanoparticles were taking the lead in recent times, which mediates us to the stimuli response therapy also. And this stimuli response therapy is majorly of two kinds. One is endogenous and one is exogenous. In endogenous stimuli response therapy is inside our body. It's regarding our pH and enzymatic responses, which takes the lead for delivering the drug into the specific site. And exogenous is from the outside of the body, which we give in the kind as like ultrasound, magnetic field, redox reaction, and photothermal therapies. And my topic here is ultrasound regulated system. And as all we know, we take ultrasound is like a cost efficient and to treat and to diagnose the disease for imaging mostly. And this in ultrasounds mostly consists of acoustic waves with the frequency which is above the human being here, and the pressure which we apply here is directly relates to the amount of the pressure we give to the targeted tissue. And here the main physical properties were like authentication, reflection, refraction, amplification, absorption, and scattering. And here in biological purpose, ultrasound is mainly of three frequencies, mainly of three kinds. One is lower frequency, medium frequency, and higher frequency. There is a saying that higher frequency leads to the higher resolution, but it has gone to the low tissue penetration, as well as low frequency will give to the limited resolution, but it will go to deeper into the tissue penetration. And we measure with the milliwatt per centimeter inverse. As a low frequency, we majorly use for superficial tumors like skin, head, and gynecological carriers, and high frequency we used for the destruction of tumors with the focal point present exactly. But when we give to the high frequency, the normal tissues were also getting harmed. So here we are going to use nanosensitizers. When we give nanosensitizers to the particle and we load the drug to it, when we apply the ultrasound, the heat produces and the drug will be released at the specific part. And most of the ultrasound response are polymer-based drug delivery, and it consists of microbubbles, nanobubbles, nanoemulsions, nanodroplets, measles, and vesicles. And microbubble and factors that affect is molecular weight distribution, that polymer shape, polymer structure, degree of polymerization, supermolecular assembly, heterogeneous interface, and bond strength. As we microbubbles were the already FDA approved some of the microbubbles, as it gives the high intensity with the endogenous microbubbles, which behaves like a cavitation, which creates high gas pressure and the temperature, and it mostly used to give for you a ultrasound mediated gene of drug delivery. And microbubbles can go into blood-brain barrier without damaging or losing the tight junctions of the capillaries. And here is the structure of a gas bubble, like outer shell and inner core. In microbubbles, inner core will be the gas and outer shell will be the polymer or drug what we are going to load. And we have some many disadvantages using this microbubble, such as it has a large size compared to the nano, and it has high osmotic pressure and blood vessel dilation. To overcome this, we are going to the nano droplets. In nano droplets and nano bubbles were the kind where we use polymer coat and core will be the gas, and nano droplets polymer coat and liquid will be the core, and it is mostly composed of perfluorocarbon. And then combined to nano emulsions. In nano emulsions, we use like two invisible liquids that stabilize amphiphilic copolymer as surfactants using nano emulsions, or phase change contrast agents that leads to drug delivery of their longer recollection time. Most importantly, all components of nano emulsions are currently widely used in clinical trials, and these polymeric PFC nano emulsions might be promising clinical transferable platform for ultrasound responsive drug delivery. And polymer vesicles and micelles, and as the here temperature, self-assembly temperature is less than the transition temperature, measles will having no impact, as well as vesicles will be having a poor impact in ultrasound response. When when self-assembly temperature and the transition temperature is equal, the vessels will be the highest, excellent ultrasound responsive, and measles will be normal state. And when when transition assembly temperature is greater than transition temperature, and the particle will be stable. This is the mechanism where the cavitation occurs when the thermal effects, and the applications will be mostly with the increased cargo delivery of the cell permeability, control cargo release, ultrasound imaging, contrast, and photometric imaging. Like we have ultrasound responsive drug delivery, where we combine 2 or 3 drugs together with the help of polymer. Many of the scientists have literatures has recently published few articles on this. And my conclusion, like there are many limited studies on this ultrasound responsive drug for the major cancer therapy, and this
Okay, Gautam, go for it. If you have any,
I acknowledge my team for this. Thank you.
okay, thank you so much. Dear participants, we did not be able to give you the time for the discussion. The participants are here. We can discuss with them during the lunch break. So all are requested to be assembled here at 2:30 p.m. for a lunch interruption. Thanks a lot for your kind attention.
Now the lunch is provided in the time, so please go and have lunch. Thank you. talk. The presenter here is Professor Luke Elizabeth Hanna from ICMR, National Institute of Research and Tuberculosis, India. So please introduce her.
Good afternoon, everyone. So today we'll be having our keynote address delivered by Dr. Luke Elizabeth Hanna. She is Scientist F and HOD of the Biology and Biotechnology Department in National Institute for Research in Tuberculosis. She has completed her BSc in Zoology from Stella Maris College, MSc in Biomedical Genetics from Dr. A&M Postgraduate Institute, and PhD from NART, Chennai. She has also a postdoc from Albert Einstein College of Medicine, New York. Dr. Hanna is from a immunology background and has experience of more than 20 years in research. She has also backed many research grants, including ICMR, DHR, PDT, and NIAID, and has also successfully completed many research projects. She has more than 120 publications in various international journals and more than 25 conference proceedings, and more than 5 book chapters. It is a great honor to have you here.
Good afternoon to one and all. So I would, before I start my talk, like to thank the management of Crescent College and also the staff of Biotechnology Department for having me at this meeting. So I know it's a little difficult to concentrate soon after lunch, but so I'm just briefly going to go through a lot of things. What is actually the key to good science is that excitement about science and the curiosity. So from all the things that I tell you, it's enough if you catch some excitement and also know that there is a lot to explore in the field. That curiosity will keep you moving. So I will just move on with my talk now. So I'm going to briefly talk to you about the applications of nanotechnology in the field of HIV/AIDS. So just to tell you about the disease, we know that the HIV pandemic has been around for 4 decades now. The disease was first identified in the year 1981 in a group of gay men who presented with a typical type of pneumonia. So subsequently, a month later, there was another report also among gay men who had a different or a rare type of cancer called the Kaposi sarcoma. So that time people realized that this was a kind of immunodeficiency disease, but they thought that it was unique to the gay population and it was known as the GRIT or the gay associated immunodeficiency syndrome. But very soon after, people started observing similar conditions, sorry. Continue. Yeah, so people started noticing this condition also among non-gay population, that is among children, adolescents who had received the blood transfusions, women, and then it came to be known that it is not restricted to the gay population, and the name subsequently got changed to AIDS or acquired immunodeficiency syndrome. But it was only 2 years later, that is only in 1983, that the person that you see on the slide, his name is Luke Montagnier. He worked at the Louis Pasteur Institute and he identified that a retrovirus called the human immunodeficiency virus was the causative agent for this particular condition. And for this, he bagged the Nobel Prize along with the person next to him, who was also one of his former students, and her name is Barry Sanosi. How do I move this slide? Okay. All right. So now we are like 4 decades after the discovery of the virus, and we have traveled quite far. There has been a lot of research which has gone into the field of HIV to control the pandemic. But where are we now? If you think about that, we still do not have a cure for HIV. HIV is still not a curable disease in spite of all the research and investment that has gone into the research for controlling or preventing HIV infection. We have so far achieved or seen only 2 cases of cured HIV, and this also is not a sterilizing cure. We call it functional cure, where there is suppression or sustained suppression of viral replication within the person's body. So for all other instances, a person who is HIV infected needs to be on treatment lifelong. Also, we don't have a vaccine that can protect against HIV so far. So these are obviously major challenges, and this says that we still have a long way to go before we can say that we have the HIV pandemic under control. But we have progressed quite well with regard to developing strategies for intervention or preventing the transmission of HIV infection. So we have made significant progress. We have been able to achieve significant decline in mother to child transmission of HIV infection, male to female transmission. We have also made significant progress in the development of pre and post exposure prophylactic methods. There is also preventive strategies such as voluntary medical male circumcision, and most importantly, the development of antiretroviral drugs to treat HIV infection. So just to bring you all on the same page, I just want to quickly go through the HIV life cycle to tell you why it is so challenging to deal with this virus. So this is a very small virus. It is only about 120 nanometers in size. It has just got 9 genes, and still it is offering such a huge challenge to the research community. This is because it has got a unique type of life cycle. So the life cycle is shown here. You see the virus, which tries to look for cells expressing a particular receptor, and the major receptor for HIV is the CD4 molecule, which is expressed on a subset of T lymphocytes known as the CD4 positive T cells or the helper T cells. When it finds such a receptor, it binds to it. There is fusion between the viral and the host cell membrane and the entry of the viral core inside the cell. Once the viral core reaches the cytoplasm of the target cell, there is shedding off of the protein code. The viral genetic material, which is RNA, gets reverse transcribed by HIV's enzyme into a complementary DNA molecule. This complementary DNA is transported inside the nucleus of the target cell, and it is integrated into the host genetic material, becomes an integral component of the host genome thereafter. And at this stage, the virus becomes referred to as the provirus. And here is the challenge. Till date, we do not have any method or strategy to eliminate the integrated provirus. So when there is an integrated virus within a cell, it remains there as long as the cell lives, and it can continue to produce viruses. So the advantage that HIV has by employing this mechanism is that it can exploit the host cell's transcription translation machinery to make more viral RNA and viral proteins, which can assemble and make more viral particles, which can bud off from this infected cell, mature, and go on to infect other cells. So here I'm showing you the strategies that we have for blocking HIV replication. So as I mentioned, we don't have any drugs which can cure HIV, but we do have drugs which can prevent the multiplication of HIV or the spread of HIV from one infected cell to another. So these are the different classes of drugs which are all shown in those yellow boxes here. So you have drugs which can block the virus from entering into the host cell. So you call that class of drugs as entry inhibitors or fusion inhibitors. Then you have classes of drugs which can target the enzymes of HIV. You have reverse transcriptase inhibitors, integrase inhibitors, and protease inhibitors. And all of these are key enzymes which have important roles to play in the HIV life cycle. And if they are blocked, HIV replication and maturation does not occur. So these are the only available classes of drugs that we have. And the problems that these drugs have are that they are not able to completely eliminate the integrated virus, for one thing. And secondly, these drugs are not able to reach certain regions of the body or the tissue sanctuaries where viral reservoirs are there. So when I talk about viral reservoirs, these are all right. So viral reservoirs are nothing but long-lived cells. Okay, so these are cells which live for several decades within the human body, and if these cells happen to get infected with the virus, then they are able to hide within tissue reservoirs and capable of producing viruses over a period of time. So these are the major challenges which the currently available ART is not able to tackle. So now we are in the era of nanotechnology, and that is what this 2 days conference is about. You would have seen about a lot of applications that nanotechnology has got. Here are some of those. And one of these applications, of course, is its role in medicine. So in medicine, it has got again several applications such as in imaging, diagnosis of disease, drug delivery and treatment, as well as gene therapy and genome editing. So in the context of HIV, I'm going to talk to you for the next few minutes how nanotechnology has been exploited to address certain issues which offer a challenge to the control of HIV infection. And we are going to look at its role in HIV treatment, in latency reversal, its role as vaccine candidates, and also for gene editing and preventing HIV infections. So, why are nanoparticles or nanotechnology, why has it become such an important technology? Because these nanoparticles are so very interesting. They have a number of unique properties which other molecules and compounds do not have. And so these can be very well exploited for human use. So these are some of the features. Probably you have heard about all these characteristics and features during the series of talks that have happened here. But what is very important is that they are biocompatible. They do not elicit any immune response within the body, so they can be safely used for human administration. So they are also easily synthesizable. They are quite cheap, so it is also quite useful for treatment or other approaches that we would like to use them for. And they have certain physical and chemical properties which make them suitable for biomedical applications. And more importantly, they do not accumulate within the body. They can be easily degraded or excreted from the body, leading to reduction in toxicity. So now I'm coming to the actual applications. So firstly, we are going to look at their role in the treatment of HIV infection. So here there is a term called laser ART. So that laser here actually stands for long-acting, slow effective release ART. So this means ART is the antiretroviral therapy or the drugs which are used to treat HIV infection. And as I had just mentioned, people who are HIV infected need to be on treatment lifelong. It is not a course of treatment which can be taken and discontinued. That is because once treatment interruption happens, all these latent viruses in the tissue reservoirs, they will start producing new viruses and there will be viral rebound. So the person has to be completely and continuously covered under ART. But obviously the antiretroviral drugs have their own toxicities, side effects. They can interact and form toxic intermediates, and they are also costly because you have to take a number of drugs over a long period of time. So to overcome all these challenges that the current ART has, we are now looking to nanotechnology and trying to see if we can make use of this particular approach for treating HIV infection. So when you have long-acting, slow-release drugs, you might have heard about that in for other diseases as well. So the advantage is that you don't have to give daily doses. You can reduce it to bi-weekly, you can reduce it to monthly dosages, depending on how effective your compound is in the slow and sustained release of the drug. So here people have tried making such slow release compounds, and one of these is the lamivudine, which is which is used in the first line treatment regimen for HIV infected individuals. So they have made a nano crystal from this drug, and they have demonstrated that this nanoparticle, which is conjugated with the NRTI class of drug, was able to reach different parts of the body, such as the liver, lymph nodes, spleen. So it could reach even the parts of the body where the normal drugs do not reach, and they continue to remain there for up to 30 days. So which means that you can reduce the dosage or dosing schedule to monthly instead of a daily. What does this, how will this help? This will reduce the toxicity of the drug that we are using for treating HIV infection, and also it will increase adherence or compliance. It's very difficult, for a person to every day take treatment. It becomes sometimes because of the side effects, a person will have inhibition to take the treatment. Sometimes it can be, it become boring to every day take treatment. So when you have certain long duration treatments, it becomes very imperative to look for shorter treatment options or less frequent dosing options, and that is where this becomes very important. So there are several other molecules or nanoparticles which have been developed for treatment of HIV infection. So one that I had mentioned was with one particular drug, that is the lamivudine, but there are subsequently there have been efforts to make also combination ART, because HIV is never treated with a single drug. It is always treated with 3 or more drugs belonging to at least 2 or more classes. So when you have to treat them, you need to have with nanoparticles, you also need to have nanoparticles which are conjugated with more than one drug. So combination of 3 drugs have been made, and these have been tested in primate models, and they have shown good in vitro as well as in vivo efficacy. So these the different drugs have been tried from different classes, like the RT inhibitors. Lopinavir is a protease inhibitor class of drugs. Then we have the integrase inhibitor class of drugs. So with all these classes of drugs, various nanoparticles have been developed, and they have been tested in animal models, humanized mice, as well as in vitro models. And there is also one combination which has been developed for the first line ART drugs, which has been also tested in an in vivo model and showed enhanced bioavailability, improved PK profile, and diminished cytotoxicity, which they want to test in a clinical trial. So moving on to the role of nanotechnology in latency reversal. So I just mentioned how important it is to, this concept of latency is in HIV infection. So unless you get rid of all the latent reservoirs, you can never come up with a cure for HIV. How do you get rid of these latent reservoirs? So latent reservoirs, as I mentioned, are nothing but cells which are not actively doing protein synthesis or proliferating, replicating. So these are all resting cells or silent cells. So because of this silence, there is no multiplication of the virus also happening within the cell, because the virus is actually depending on the host cells, enzymes, and proteins to do its own, produce its own RNA and proteins. So when viral replication is not happening, viral proteins are not being made, the cell is also not expressing the viral proteins on its cell surface. So it is not detected by the immune system as an infected cell. So it is continued to, so it continues to remain within the host immune system. So it actually escapes the immune surveillance which is happening. So in this picture, you can see when a person is infected with HIV, he has a lot of active cells in the body, activated cells which are doing certain functions. So all these quickly get infected with the virus. But there are also some cells which are not very actively proliferating or functioning. And these cells are few in number, but these cells are long-lived cells. So they travel to remote parts of the body, lodge themselves in tissue sanctuaries where they are protected from the entry of drugs. So these are the cells which will remain when a person is on treatment. So we are able to tackle the virus which is getting produced and multiplying. So that is taken care of. However, the virus which is lodged in the reservoirs continues to remain. So when the person is off ART or when there is treatment interruption, there is actually a rebound effect, and you see a lot of green cells which are coming back into circulation. So when you want to completely cure this person of HIV infection, what you need to do is, when you have the person covered on ART, there are no virus is completely under control, no extensive viral multiplication, only those reservoir cells are there which are few in number. So if you activate these cells and start making them to produce virus, then the immune system can recognize those cells and clear them from infection. And the agents which are capable of doing this are called the LRAs, or the latency reversal agents. There are several classes of latency reversal agents which have been identified. The most popular ones are the HDAC inhibitors, and certain pro-inflammatory cytokines, as well as the protein kinase C activator. But drug-loaded nanoparticles also are now becoming popularly used as latency reversal agents. So when these nanoparticles get loaded or conjugated with latency reversal agent, they not only can travel to these remote sites because of their small size and enhanced solubility, they also can be targeted to specific cells. Like we know that these are long-lived cells, memory cells, which are more prone to become latent reservoirs. So these can actually be targeted to those cells and activate them to make more virus, thereby making these cells visible to the immune system and pave way for viral clearance from the body. So these are some examples of nanoparticles which have been developed as for the purpose of latency reversal. So some of them are only nanoparticles which are conjugated with the latency reversal agent, but some of them are also loaded with or encapsulated with the anti-HIV drug. So they do a combined function. The latency reversal agent goes there and activates those cells. The cell starts producing the virus, and the antiviral drug, which is also encapsulated within the nanoparticle, now takes care of the virus and prevents it from multiplication. So there are different platforms, nanocarriers, which have been used for carrying these latency reversal agents to the different parts of the body, thereby making it useful for inhibiting viral spread and viral latency. There is also a role for nanoparticles in or nanotechnology in HIV diagnosis. So HIV diagnosis is actually very easy, and there has been a lot of progress in this line. So we have very simple rapid tests which can give HIV results in about 10 minutes. But the only problem here is that these tests are all antibody-based tests, which means that they depend on or they detect an adaptive immune response to HIV infection. So you might know that for a body to develop a robust adaptive immune response, it takes a few weeks. So it could be easily 3 to 6 weeks before you can find detectable amount of antibody within an infected person's body. So antibody tests have this challenge that they cannot detect HIV infection in the very early stages. So within the first 3 or 4 weeks of infection, the antibody tests do not perform well. So are there alternative tests? Yes, you can look for antigen. So antigen detection ELISAs are available, but there again, it is most useful only in the 2nd or 3rd week after infection. But if you want to detect infection much earlier, the obvious test that you have is only a nucleic acid based test, where you can detect either the HIV RNA or the integrated proviral DNA to establish HIV infection. So the nucleic acid tests, although they are now become commonplace, still do have certain requirements and certain challenges. It is difficult to make it a point of care test. It requires certain infrastructure, facilities, equipment, expertise, and so on. So it would be very useful to have more simple point of care tests which can really be used at at the field level to give rapid test results. So nanoparticles, particularly gold nanoparticles, have been exploited for this application, where they have been developed as nano sensors, which have high sensitivity and are capable of detecting even nanogram and picogram concentrations of HIV antigens for HIV diagnosis. So it is believed that within 2 to 3 days of infection also, that amount of viral antigen, which is circulating protein, which is circulating in the system, can be detected using these highly sensitive nanosensors which have been developed. So now moving on to vaccines, I told you we do not have any vaccines for HIV so far, and the challenge obviously is because of the high genetic diversity, which is a characteristic feature of this virus. So the virus keeps on mutating, changing its structure, so you cannot have a universal vaccine which can protect against HIV. So you come up with a vaccine candidate and you vaccinate a person, the person's body starts making antibodies. The virus sees it, it quickly changes its epitope or the antigen. So the antibody is no longer able to neutralize that virus. The virus continues to infect and multiply. So apart from that, that is a major challenge here. So for all vaccines, we have certain characteristics which need to be fulfilled for nano vaccines as well, meaning that it has to be safe, it has to be stable, it has to induce a potent immune response within the body, it should not revert back to a virulent form, and there should be good not only uptake of the antigen but also presentation to the cells which are actually responsible for initiating the immune response. So there are several vaccine candidates which have been developed for HIV based on the nano platforms that are available, and some of these are listed on the slide. So here the first one is actually an interesting candidate where they have conjugated HIV proteins P24 and the NEF, which are both HIV antigens. These are conjugated with a toll-like receptor called TLR5, and this has been coated on the surface of a lipid based nanoparticle. And the advantage is it has been demonstrated that these nanoparticles are able to identify antigen presenting cells, home onto them, present the antigens, and initiate a robust immune response. So there are also ferritin based nanoparticles available which allow the vaccine candidate to actually home into the lymph nodes, activate the germinal center, and amplify the immune response which is required. So there are also nanoparticles which are targeted to Langerhans, which are again antigen presenting cells, and there are a number of candidates which are also able to initiate a broadly neutralizing antibody response. So when we talk about antibodies have got several roles to play. They can opsonize, they can help in phagocytosis, they can help in apoptosis, but there is a subset of antibodies which are known as broadly neutralizing antibodies, and their role is actually to bind to the virus and prevent it from binding to a target cell. And these broadly neutralizing antibodies are very important for a preventive or a protective HIV vaccine. So there are some candidates which have been developed which can potentially generate broadly neutralizing antibodies within the host immune system. So I think I'm running out of time. I'll take the next 5 minutes to quickly say a few more things. So here we are coming to viral fusion, which is, as I told you, the very first step in the HIV life cycle. So we have silver nanoparticles, we have hydrophobic polyacrylate based nanoparticles, and these are all able to block the infection process, that is the fusion process, or the first step in the HIV infection cycle. There is another phenomenon called biomimicry, where you try to mimic a normal human cell through a nanoparticle. So here we have nanoparticles which are coated with the CD4 cell membrane, T cell membrane. So when the when the body looks at it, it will appear as a CD4 positive cell. So this has an advantage of neutralizing the viruses which enter and reducing viral infectivity. There is something called extracellular vesicles. So these are not actually nanoparticles, but they are nanoscale, naturally occurring subcellular components, and they have been identified to have an important role to play. So this person called Palmino et al. Recently found that extracellular vesicles, which are released by lactobacillus, which are common commensals in humans, they are able to prevent HIV infection of cells. So this gave an interesting clue to a group called Welsh who took this up, and they also started isolating extracellular vesicle and looking for their activity on HIV. And they found that these vesicles isolated from the semen of HIV infected or uninfected individuals could actually inhibit HIV infection. So there is a potential for this particular technology, EV-based nanotechnology approaches in preventing the spread of HIV infection. So I have to say this because, now this is another technology which is like, having a lot of applications in various fields, which is genome editing, and you might have heard about a number of gene editing. And one of these tools is called the CRISPR-Cas method, which has become the most popular and most efficient gene editing platform currently available. And this is also being exploited for the treatment of HIV infection. So gold nanoparticles conjugated with CRISPR components have been used for doing editing of the hematopoietic stem cells so that these cells become resistant to HIV infection. So this is all towards gene therapy, where also nanotechnology is finding its place. All right, I'll skip this slide. And nanoparticles have a role to play in enhancing or boosting the innate immune response. It has got a role to play in blocking all the enzymes of HIV, which I spoke about: HIV reverse transcriptase, integrase, and protease. And also another important characteristic is that it can penetrate the blood-brain barrier. So we know that the brain offers a large anatomical barrier, but HIV is able to enter the brain, infect the macrophages which are present in the brain, known as the astrocytes and the microglial cells, and the blood and the drugs, they do not very well reach these sites, so they continue to multiply there, and because of this they can result in neurocognitive diseases. So to prevent this, there have been the nanoparticles and nanoplatforms have been exploited to cross the blood-brain barrier and deliver the drugs inside the unreached brain of the infected individual, so as to reduce HIV replication there and also the consequences of HIV infection. All right. So finally, in HIV prevention, so I already told you in one of my first slides that we have several methods which are available for protecting against HIV transmission. And here we have gel-based nano emulsions, which are also developed and have shown very good protective efficacy, both in animal models as well as in humans. And they are also being used for pre-exposure prophylaxis. All right, so I have come to the end of my slide. I'm sorry if I have rushed so much, but actually I'm just concluding with what I started. So I have just placed before you the wide range of applications that nanotechnology has got in this small field of HIV infection. And if you're excited about this, you can always go back and read. So I'd like to thank you for your attention, and I would like to thank the organizers once again for this opportunity. Thank you. If you have any questions, I would be happy to take.
Thank you so much, ma'am. Thank you for the informative talk. Since we are running out of time, there is one question is allowed. Any one question?
Hello, madam. So you have given very nice presentation. In your presentation, you just mentioned the gold nanoparticles properties, the surface plasma resonance tube. After that, you mentioned the magnetic properties. I don't think gold nanoparticles have magnetic properties.
For gold, yes. Right? Sorry.
Yeah. And the next one, what about the selectivity? So when we are checking the HIV, other viruses also will be present. So did you check the selectivity of your kit?
Yes. So actually, all these antigens which I kept mentioning there, HIV1P24, HIV1F, HIV1RT, all these are highly specific only to this class of viruses. So some of these, like the reverse transcriptase, they are present in all the lentiviruses. However, the HIV1RT is very specific also. So it performs selective inhibition, but there are some many of these gold silver nanoparticles, they have intrinsic antiviral activity, so they can also act on other viruses. But when you really want to do targeting, you can still go ahead and do selective inhibition.
Thank you, ma'am.
I welcome Professor Dean
Thank you.
Rahman to felicitate. Thank you, ma'am. Validity session is about to begin.
Yeah, got it, sir. Deep sync.
Good evening, everyone. We now have come to the end of the 2 days international conference on applications of nanotechnology in biology, biotechnology, and biopharmaceutics. I now request Mr. Bashir to inaugurate the valedictory function with Kiran recital.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم لإيلاف قريش إيلافهم رحلة الشتاء والصيف فليعبدوا رب هذا البيت الذي أطعمهم من جوع وآمنهم من خوف صدق الله العلي العظيم. Translation: In the name of Allah, the most gracious and the most merciful. For the accustomed security of the Quraysh, their accustomed security to the journey in the winter and the summer. So they should worship the Lord of this house, who has fed them against hunger and made them secure from fear. Amen.
Thank you, sir. I now request Dr. Karthikeyan, Professor and Dean, Student Affairs, to deliver the welcome address.
So, good afternoon and have a good day one and all assembled here. So, dear colleague, professors, lecturers, researchers, and ladies and gentlemen, on behalf of Crescent Institute of Science and Technology, I would like to express my sincere gratitude and welcome to you to the Indo-US International Conference on Applications of N3, Nanotechnology and Biology and Biomaterials, 2022. So moreover, I am honorably welcome our Vice Chancellor and also valedictory function invited guests of the session. This conference helped the students to get into a self-introduction as well as their achievements. So this encouraged the students to participate in various events and see that blossom into the individuals. I would also like to thank our Dean, School of Life Sciences, for all the enthusiasm, energy she carries for the towards the students and all the participants. It's my hope that this conference was achieve these objectives by providing effective forum for the academicians and researcher and advancing the knowledge, research, and technology for the humanity. The focal drive of this conference is the exchange of ideas and participating in this exchange. It hoped that all the parties benefited from the conference and can apply it is managing the activity in their areas also. It is pleasing to note that this agenda of this conference covered a wide range of interesting topics related to all the theoretical, practical aspects of the sciences. And last but not least, my deepest gratitude goes to advisory boards, organizing committee, international scientific committee, institution, companies, and volunteers who directly and indirectly supported this successful conference. The committee has organized a vibrant scientific program and this worked hard to present highly respected and internationally known speakers to lead it. Although we tried the finest to this professional on behalf of BS Abdur Crescent Institute of Science and Technology, we accept our sincere apology should be there to inconvenience if that occurs before or during or after the event. I hope you had a very productive conference with exciting and encouraging discussion and exchange of knowledge, so that together we can anticipate future groundbreaking knowledge in the research and technology for humanity. So may God bless all with good health and to make this conference very successful and enjoyable one. And again, once again, I welcome all of you for this wonderful valedictory function. And also we are thankful to all of you who spend all 2 days with us. Thank you so much. Thank you.
Thank you, sir. I now request Dr. P. Mohammed, Vice Chancellor, to present the presidential address.
Good afternoon, all of you. Dr. Dharani Gopal, the chief guest of today's function. Dr. Hemalada, Professor and Dean, School of Life Sciences. Dr. Karthikeyan, Professor, School of Life Sciences. Faculty members of the School of Life Sciences. And the students from the University of East London. My dear student friends. So we have come to the conclusion of this Indo-USA International Conference on application of nanotechnology in biology, biotechnology, and biopharmaceutics. As I understand from Dr. Karthikeyan, there were a good number of papers on these topics for the 2 days. And of course, you would all enjoyed the delivery of the paper, the discussion, the deliberation. Definitely, it should have been more engaging, enlightening for you to gain more knowledge and to widen your horizon of your understanding. Of course, the knowledge cannot be limited to one particular area. So to deepen and widen, you should interact. So the better forum for interaction is a conference like this. You'll come to know about your peers working in the related areas, which will help you to network further. That's most important thing. Please take this point very seriously. So the collaboration is a must for coming out with any useful innovation. It's not that an individual can deliver, never is possible. Might have happened some 200 or 500 years back. Now, without collaboration, you cannot make anything useful because the knowledge is very wide, and you develop interaction with the people who work in the related area of your research and continue your interaction. Do collaborative research work. You visit their place, and you can also invite the experts to our place. So that kind of exchanges is a must. Physical and of course knowledge-based exchanges are very, very essential. So I congratulate Dr. Hemlada and her team in organizing this kind of wonderful international conference for the benefit of researchers, students, and academicians. So those who organize the conference would have known that it's not an easy job to organize a conference of this nature. A lot of efforts, works should have happened actually behind the scene. What we see may not fully explain what are the hard work they have undergone. So I really appreciate all the efforts taken by all the faculty and professors concerned in making this conference a grand success. Similarly, I appreciate and of course congratulate the completion of your 6-week program by the students of University of East London. Actually, it's an honor and privilege for us hosting us in our campus. I'm sure that you would have definitely enjoyed the hospitality extended to you in all aspects. The stay, of course, in the learning experience, and visiting the different places and coming to know about our country and particularly our people in particular. So that actually has made a lot of effect on you. I could understand that you adapted our culture. You all come on with saris and dhotis. Really, it's a reflection of your love and affection towards our culture. We appreciate a lot. And this kind of exchanges, as I told that, isn't it? In UK, isn't it, particularly University of East London, and compared with our university, maybe advanced in their stature. Still, they want and learn from us. So that shows the importance of exchange of ideas. It's not that knowledge is limited to one particular place or one particular university. No. We have to travel far and wide and to get the knowledge, information, experience, everything. Likewise, our students also should visit different places. It's not that advanced countries, any places actually. So to come to know what are the problems existing and to find some solution from the knowledge you have gained, particularly in the area of biotechnology, I would say that. So biotechnology is leading the way in all aspects. People are talking about green economy and organic foods, all related to biotechnology. I used to say that earlier, the economy developed based on chemical processing. In 50s and 60s, particularly Chennai, I would say that. Chennai was basically a chemical-based city. Whether you visited Manali and Ennur, you could see a lot of chemical industries, fiber industries, fertilizers, oil, so many large-scale industries are available there. And Chennai developed from the chemical industry. Now people are moving from chemical to bio. So fortunately, you are on the right track and the right field. So you can lead the way, and there are a lot of opportunities. So to make our life healthy, comfortable, secure. So you could give the right solution to our people, the biotechnologists in general. I'm talking about that. So it would be the order of the day from now, actually for more years, 10 or 20, 30 years, people will talk about only biotechnology, bioprocess and other things only. So this kind of conference definitely help in improving our exposure and gaining more knowledge. And with that, definitely our standing would improve and our contribution to the society would be better. So finally, I congratulate again the Dean of the School of Life Sciences and a team in organizing this conference and hosting the students from the University of East London, and every way we benefited. I wish that similar programs would be organized in future by the department for the benefit of everyone. Thank you all.
Thank you, sir. I now request Dr. S. A. Madatha, Professor and Dean, School of Life Sciences, to give a report on Turing program.
Good afternoon to all of you. I am really happy to see all of you again after this inaugural function. I am here to give the report on the Turing Grant program. You could have seen all the students. They're all here from University of East London. We could have interacted. And we have started this 6 weeks program on July 4th. And this journey started in 2015 when the British Council of Biotechnology mission was presented by the delegates from various universities of UK. And from that, we have initiated our collaboration. And we have signed a MOU with the University of East London on August 2016. And then we have started research collaboration with them. And also we have jointly organized international conference in 2016. Then we have got the grant to work on novel antibiotics to tackle antimicrobial resistance. It's an international project between UK and India. And then we have initiated this international exchange program that's funded by the Turing Grant. And this Turing grant is funded by the government of UK. And there are several applications, like thousands of applications. And in their university also, there were 170 applications. Out of 170 applications, only 6 were selected and they came here. And it's a very, very good installation. And they came here on 3rd July, and then they have been with us until today. And then their program is ending today, and they'll be leaving to UK tomorrow. And this is the inauguration of Turing Grant, and this was done on 4th. And this is also inauguration. And this program includes lectures by various faculty members, and all of our faculty members delivered various lectures on various areas of biotechnology, nanotechnology to the Turing students. And also we have organized interdepartmental lectures, and the different faculty from various departments like computer science, chemistry, then mechanical, civil, they also delivered lectures and they have interacted with the UK students. And they also visited various schools and departments of our university. And this is the sample picture of their work. They are working in the nanotechnology project in labs. And this program also includes various industrial visits, and this shows their industrial visits. And also they have interacted with higher officials like US consulate officials, as well as with the chancellor, advisor, vice chancellor, etc. This program also includes heritage and cultural tours, and you can see here their heritage tours, their cultural tours. And I'm happy to say that even though they are spending their weekends in the cultural tours and heritage tours, they are very serious when they are working in the labs and when they are attending the classes. And I am really appreciating them. The very good outcome came from these projects. And this is one of the outcomes. They have attended the quizzes and they have won within quizzes. And they have presented papers yesterday in this international conference, and they have generated very good data. And I'm happy to announce that they have returned 6 manuscripts individually. All of them have returned the manuscripts and also 6 review articles. And they are all ready to be communicated, and we'll be communicating either this week or next week. Because of this conference, we are really busy. We couldn't communicate on time, and we'll be communicating by next week. I really thank all of you. And let's acknowledge our management, our Vice Chancellor, Registrar for supporting us and also for their encouragement. And I thank our Senior General Manager, Mr. Jalal, for providing separate book for them and also separate vehicle for them to roam around. And also I thank all the faculty members of School of Life Sciences, research scholars, and also the technicians, and also other faculty members from other departments, deans and HODs for supporting this program. And I'm thankful, and special thanks to Dr. Wasim Malai. He has been with the students, and he is from chemistry department, and he has been working with them. They have developed very good data, and he has been teaching all the analytical techniques, then analyzing the data, and it's his project, and then he has played a major role in developing this project. And I also thank all the hostel people of both men's hostel as well as ladies hostel, and all the people who are directly involved and indirectly involved. Thank you.
Thank you, ma'am. I now request the dignitaries to honor the faculty members involved in Turing program. I would like to invite Dr. Haji Sheikh Mohammed, Dean Academics. Dr. Aisha from Computer Science. From Chemistry Department for giving special lecture as well as mentoring the students with project-based learning. Dr. Irfan from Pharmacy Department. Architect Tilagavadi ma'am. Dr. Karthikeyan. Dr. Ashok Kumar. Dr. Krishna. Dr. Nisar Ahmed. Dr. Shazia Jamal. Dr. Sangeeta. Dr. Sangeeta. Dr. Mubarak Ali. Dr. Farida Begum. Dr. Simon Duregas. Dr. Bhaskaran. Dr. Nasir Hussain. Dr. Shanbaj Khan. Dr. Fazila. Dr. Ranjini. Ms. Narmada.
Ms. Sabna. Ms. Archita.
I'd like to ask the UK students to give the feedback on Turing program.
Okay. Hello, hi. Hi everyone, hi. So this 6 weeks has been extremely interesting. I've learned so much about nanotechnology and nanoparticles, which is all very new to me. This university has been very accommodating. With all the help of all the professors have been fantastic. They've been so patient with us, even through the stressful times. They've supported us, taken their time out of their schedule to really put in the work to educating us. And yeah, just special thanks to everyone, and of course the students as well for supporting us. And yeah, I have a lot to say, but big thank you, and this has been a wonderful experience, really interesting. The food is delicious. Your country is absolutely beautiful. Thank you.
First of all, I'd like to say thank you. This was an amazing opportunity. Thank you to all the teachers and to Rahman, the university. At first, I was really nervous before I came, and as soon as I got here, the hospitality of the teachers, students, and Indian people has been absolutely overwhelming. I'm so glad we got the opportunity to undertake new projects and potentially get published. So I'll remember this for the rest of my life. I'll definitely be coming back to India and tell everyone to visit.
Hi everyone, my name is Enrica. So I'm really grateful I came here to do this experience. I learned a lot of nanotechnology, about the people here, about myself, and it's been amazing. So thank you to everyone, to the teachers, everyone that organized, the people in the hostel. So thank you very much for this amazing experience. Thank you.
Hi everyone. Since I came here, everybody said that I look Indian, and I think today I really look Indian. Thank you very much to everyone. Especially to Dr. Gangani and Dr. Himalato. They've been amazing. Like, I feel overwhelmed with the love I receive in this country. You all should be always proud of your culture. They're amazing. And I think I have stories I will tell to my children on the road.
Hi everyone. It's really pleasure to meet you all. A very big thanks to the Dr. Himalato as well as all the other staff who help support each and every circumstances we face. And I'm really thankful to the Abdur Rahman Institute of Science and Technology. And when I go back to London, I'll tell my friends, family members, friends, to everyone about this experience. And also I'll tell my juniors as well in my university that I'll promote to each and everyone that this type of program will continue forever in the future in the same university. So thank you everyone. Thank you.
Good afternoon. The same, I want to say thank you to all the teachers who helped us, to the dean, ma'am, to the Ranjani, to Dr. Vasimala. I remember Dr. Vasimala was sick and he was at home, but he still was in touch with us to make sure that we complete our reports. Thank you. Yes, we are very, very lucky that I'm here, all of us. This is an amazing opportunity. I'm from a little country in Eastern Europe, and I was thinking, I'm the only person from my city who came to India, and I'm gonna go home and gonna tell to everyone I know what kind of experience I had here. Amazing, these are lifetime memories. Thank you.
Great pleasure to have you all here. Thank you for visiting us. Now, I would like to request Dr. Mubarak Ali to give a report on the international conference.
Good afternoon to all of you. Okay, I'm pleased and honored to read the conference report on the ICNB held in the last 2 days. ICNB, the International Conference on Applications of Nanotechnology in Biology, Biotechnology, and Biopharmaceutics, is a proposal is considered during the vacation July 25 this year, and immediately crystallized the idea and associated with the University of Missouri and the Biotechnology Research Institute, Microbiological Society, India, for the announcement. The last date for this conference actually is fixed August 8, and for this 2 days of deliberations. With a short period of time, within 2 weeks, the international association and international speakers are not merely a simple and challenging to conduct a hybrid mode. You know, the chess Olympiad happened last week in Chennai, they got 4 months of preparations, but our NC and we got only 4 days of preparations. So it's all happened. Without successful completion, it's not possible without the able guidance of our team. All of you participated on the campus and online are the main reason for the overwhelming responses received. Around 500 registration, 290 abstracts received from USA, UK, Malaysia, Korea, China, and all other countries. So first day, the NCO inaugurated on August 11, 9:30 in this hall. Professor Dean, the Army Secretary of this event, welcomed the gathering. The Honorable Vice Chancellor conferred the presidential address where he highlighted the importance of nanotechnology, research, and standards, even that we are initiated long before. The registrar gave a special address to the gathering and appreciated the effort taken by the School of Life Sciences periodically for the benefit of the students and the faculty. Dr. Satya Rajeshwaran, Central Council of Research Siddha, India was chief guest and delivered the keynote address on ethics in nanomedicine. It is a thought-provoking lecture and highlight the ancient practices of nano research in India. Our Chancellor of BS Abdur and Senior Manager are physically presented and grace this occasion. I myself is proposed to vote of thanks. And the first invite lecture conferred by Professor Jungwon Kim, Division of Bioengineering, Incheon National University, Korea, connected through the Zoom platform. Followed by the oral presentation were held. Dr. Karthikeyan, Associate Professor, and Dr. Vasimali from Chemistry were chaired the session. After the lunch break, Professor Mukesh Doble from Indian Institute of Technology, Madras, India, delivered lecture in campus and telecast to the online participants through the Zoom platform and also live streaming in YouTube. Dr. Srijit Ravindran from Tessa University, UK, delivered an invited talk on 3D imaging and tissue engineering aspects. Online presentation initiated with the various online halls we termed as OH 1 to 10 different halls, chaired by Dr. Ashok Kumar, Associate Professor, Khurshid Ahmed Khan, and Dr. Nazir Hussain, Dr. Bhaskar. The next today, the session initiated in the morning 9:30, where Dr. Abnesh Gangula from University of Missouri and Dr. Dhananjay Suresh from University of Missouri delivered the talk on the application of nanomedicine in retinopathy and cancer, respectively. Dr. Avinash Kumar, Assistant Professor of Indian Institute of Technology, Design and Manufacturing, Kanjipuram, to deliver a talk afterwards and highlight the antimicrobial mechanism and CRISPR technology. Dr. Sankita and Dr. Vasimile chaired the session. Oral presentation on campus continued, where Dr. Ashok Kumar and Dr. Vasimile acted as chairpersons. Online presentation initiated at 6 different online halls. These sessions were chaired by Sankita, Khurshid Alam, Dr. Nazir Hussain, Dr. Bhaskaran, Dr. Karthikeyan, Dr. Fazila. After the lunch break, Dr. Luke Elizabeth Hanna from ICMR National Institute of Research in Tuberculosis delivered an invited lecture and highlighted the nanomedicine. Now we are in the valedictory function. Dr. Karthikeyan, Associate Professor, and the student appear welcome the gathering. Our Honorable Vice Chancellor presided over the function, and Dr. Dharani Gopal, Group Head and the Scientist of National Institute of Ocean Technology, considered to be delivered chief guest and going to deliver the valedictory address. Finally, the vote of thanks will be proposed by Dr. Ashok Kumar. With that, the entire Zoom platform will be the entire event, the Zoom telecast in the Zoom platform and YouTube live streaming were handled and chaired by Dr. Simon Duraraj and Dr. Farida. The post presentation and accommodation handled by the Sanvas, Dr. Fazila. The registration and allotment handled by Dr. Fazila and Mr. Gopinath. And the timely arrangement of refreshment and lunch handled by Dr. Sazia Zaman. The scheduling and certificate guest care are taken by Dr. Ranjini and also coordinator of this event. Actually, these 2 days of the conference, we have got a special issue from the various journals. That abstract book is published with ISBN number shortly. The full paper can be submitted to the conference special issue. After the peer review process, the article will be considered. The 4 journals are consented to be published in this our conference issue or updated Biochemistry and Biotechnology from Spincher, Weight Catalysis and Agriculture Biotechnology from elsewhere, and a Nanometal Chemistry from Thailand Francis, Current Pharmaceutical Design from Penthamin Sciences. The publication and journal related information will be shared all the district participants after the conference. On behalf of myself, the coordinator, and my co-coordinator Ranjini, and organizing secretary and organizing committee of SLS, I thank, and also I thank Almighty, and we are in the 75th Independence celebration. As a scientist, we celebrate in our own way by enriching the knowledge through ICNP. So they have proven that how much we are hospitality and everything. So that is the one thing that 70% Independence is celebrated. With that, I'd like to thank once again for the active participation for these 2 days, both in on campus and also in online. Thank you so much, Jai.
I now request the dignitaries to distribute the certificates to the students of
Thank you, sir. Now we'll be having a valedictory address by Dr. Dharani Gopal, Group Head, NBT Scientist F, Marine Biotechnology, National Institute of Ocean Technology. He has completed his Masters in Fishery Biology, Zoology from Khalid Mohideen College and MPhil from New College. He has also completed his PhD from University of Madras. His research interests and expertise are zooplankton, marine ecology and biology, environmental biotechnology, bioprocess, and bioremediations. He has also published more than 30+ publications in national and international journals. It is a great honor to have you here, sir. Now I request Dr. Dharani Gopal to deliver the valedictory address.
A very warm good afternoon. Namaskar. This is Dharani from National Institute of Ocean Technology. I'm very glad to stand in front of you. It's my privilege and pleasant experience to stand. And to start with, Respected VC, Head of the Division, Dean of the Science, faculty members, students, and wonderful students from University of East London, and the Indian traditional dress. I will not take much of time. I'll take a short discussion on our work on marine biotechnology. Sorry for a short presentation, and I have grabbed some information what is available with me suddenly for this lecture. So I will be sharing my research experience on the marine biotechnology, particularly what we are doing on marine microbial biotechnology. We essentially work on a variety of biotechnology work on specific to marine, and I would like to share my experience on the work what we do on the marine microbial biotechnology. So I don't need to emphasize on ocean, you know everything about ocean, but still to make some key points. Ocean is the largest thing on this earth, which covers 71% of the Earth's surface, and we don't need to tell much. And to come to directly from ocean to us, we are also 72% of water. Every human being, every life on this earth is made up of 72% of water. Water that signify the importance of water, and the significance of planet is only the water that available in all three forms and available in liquid form. That's the sense. So the water is the sea is the water, and it's a primordial mother of everyone. And we know that, and science very well acknowledge that life originated on the sea. So what is marine biotechnology? To to harnessing the marine life for various purposes of our day-to-day activities. And it has a very huge global market at present today in comparison with chemical things. As my earlier speaker said, everything's going chemical and we are turning back to the organic form. So the biotechnology and marine is the only source where we can go organic and get back to the nature. And we have a good market. It is about $5 billion US dollars present in US alone. In 2026, it's projected. And what are the potential of a marine microorganism? It can be a whole organism, enzymes, bioactive compounds, pigments, so many things. Everything from the animal or a plant can be utilized for biotechnology purpose. To start with, the first expedition of the marine started by the first expedition in 1872 by the first oceanographer Charles from the Challenger deep on HMS Challenger by Charles William Thomson. He was the first person to go into the interior of the sea and find what is inside the deep sea in excess of hundreds of meters. So in 1875, March 22nd, the Challenger Deep Station recorded 8,184 meters. That's the first record on the deepest part of the ocean, and that paved way to a lot of work, which is today we are carrying out. And what's special about marine and what's special about microbes? The marine is one of the most oldest known biosphere, which is still existing on there. And we do not know much about the deepest part of the ocean than we know about the Mars or the land or the moon, which is several lakhs kilometers away from us. And we do not know what is beneath our feet. What is the constraint in the deep sea? It's low availability of food, no light or photosynthesis, and low O2 and low temperature. And to top of that, it's very high pressure. Every 10 meter of water add 10 meter column of the water add 1 bar pressure. And sense is we, the human being, are designed only to work in 1 atmospheric pressure. The whole system, including the respiratory circulatory system, is designed to 1 atmospheric pressure. And this animals live in excess of 100 atmospheric pressure. This has special adaptations. And the second is all we are adapted to the organic life, which is essentially supported by the solar cycles and solar energy. And this deep sea does not have those solar energy or solar cycles. It is one single day. There is no daylight or anything. It is one single season. I have picked up some of the pictures which is available already on the sites. And this is to show what is, when you see the water, it is nothing is available. But as you go deeper and deeper, where there's no light, you have plenty of organisms. It's a picture taken from a deep sea garden, which is a Gorgonian garden, and you see plenty of colors and plenty of organisms in there, which emphasize it is the deep sea is more denser and more biodiversity rich than any other biodiversity rich on the planet Earth. And this is a shot from a Precambrian Madurai, which is showing a deep sea floral garden. And in some same places, they have collected some of the sponges, which sponges, one of the very primitive organisms. It's a colonial organisms, which is very simple examples of living cells together. In fact, a human being is made up of, say, 1,000 trillion cells, and we are a consortium of living cells. Similarly, a sponge is a consortium of living cells, but each one is an individual organisms that live together, and they generate variety of substances. The first substance which we got from the sea is the spicules of sponge, which is made up of silica. And interestingly, this deep sea sponge is capable of making a meter length of silica, which is a pure amorphous silica, which is used to made in very high clarity of optics, respectively optics, which is used for research purpose and so many things. And this is one of the interesting topics where we can work, where people are working on how to mimic this silica synthesis by the deep sea organism in natural conditions. So, and there are some other very important aspects of the deep sea. This is called the hydrothermal. There are very specialized niches in the deep sea. And one of the niches is the deep sea. Deep sea is hydrothermal vents, which is in extreme conditions and where the life is supposed to be originated several million years ago. And it is completely supported by the chemical energy rather than the photosynthetic energy. and organism derives energy from the sulfur or other chemical modes. And this is one of the very peculiar organisms living in the deep sea vent, Riftia, which lives entirely on the chemical energy by symbiotic relationships. We know symbiotic relations very well in corals, but the similar kind of deep sea symbiotic, which is a chemical root, is living inside the deep sea environment. And this animal doesn't have not an usual animals, doesn't have a mouth, doesn't have a respiratory system. It completely depends on the bacteria, which sort of lives in the trypanosomes, and they support they derive energy from the sulfur oxidization, and glucose and other things are generated by this. And a lot of studies has been conducted since the identification of this organism. And similarly, there is another community called hydrothermal vent community. Is a pumpally worm, is one of the worm which is known to live in excess of say 180 degrees, 176 degree Fahrenheit, that is roughly 80 degree temperature. That means 40 degree, all the functions of this universe come to cease, some proteins will come to cease, the function will cease in 40 degree. But if you look about this 80 degree temperature living in organism, still working, still reproducing, still doing the ecological function, it is interesting. Because of the one molecule what we got from a hydrothermal, not from hydrothermal, but from hydrothermal activity, we are having whole biotechnology platform is running on a single fulcrum of one enzyme, which is doing all this process, and including today's HIV or today's COVID situation. We have that enzymes from a hydrothermal, not from a hydrothermal, from a hot spring. So with that one enzymes, we can do wonders so much we are doing. If you identify an enzyme, that enzyme will work supposed to be on, say, 80 to 90 degree temperature. But if you find an enzyme which is working on more than 100 degree, how much it will be useful for the modern days application, and to go for other applications, you must imagine. And similarly, there's another community of organisms which living in the deep sea mound, which is essentially a gas hydrate. There are a huge number of gas used, a huge amount of gas in the sea, which may be a storehouse of energy for the future. And this is a gases which in form of hydrate, and the organisms live in this deep sea. They utilize the hydrates, gas, methane specifically, and they produce the energy. So the essence of the hydrothermal of the marine biotechnology is to tackle this untouched environment, untouched chemical pathways, and untouched organisms for a variety of applications which the modern days demands. In the last 25 years of exploration, we have identified a new phylum, at least 20 new families, 90 new genera, 300 new species, and a whole lot of 250 new strains. And the maximum biomass can go up to 30 kg, which is the very highest biomass on this earth. Never you can come across 30 kg per meter cube of wet dry biomass. And it is 1,000 times greater than the biomass observed on the typical on the earth floor. Otherwise, the ocean floors are almost nil. So what we do, we National Institute of Ocean Technology, a group of scientists working in National Institute of Ocean Technology and the Marine Biotechnology, do participate in a lot of oceanographic cruises. The institute has 4 ocean-going vessels, which is an ice class vessel which reaches Antarctic often, and we participate in this kind of research vessels, and we collect on board samples for various applications and process the samples on board. So we'll go slightly to the deep sea, and we are interested in studying the microbes, and everyone is studying the microbes because microbes rules the world. And the particular microbes which is in the deep sea is one of our interests because no other people have an access to the deep sea microbes, and the pressure and the other environmental condition prevailing will not allow you to access the microbes so easily. So to go to the deep sea microbes, once you go to the deep sea, as I said earlier, the deep sea is having a constraint of pressure. It is every 10 meter, it's 1 atmosphere increases. If you go to the 1,000 meter depth, you have a 100 bar pressure. And the maximum depth of deep sea, maximum depth of deep sea is about, say, 11,000 meters. And you will have 1,100 bar pressure, 1,101 bar pressure at the deep sea. That means 1,101 bar is enough to crush any material on this earth if it is not a solid piece. So to study this deep sea, first you had to, for microbes, all of you know, because we're all biotechnologists, it's aseptic conditions required. You require to completely avoid the contamination from the surface, cross contaminations. That is one of the things for essence of microbiology. And similarly, once you go for the deep sea, the second thing that you have to negotiate is high pressure. So high pressure you have to negotiate. That means the sampler should go intact to the deep sea and able to collect the water without the loss in the pressure. Because the microbes, as we say, it is living in the deep sea can be divided into 2, generally based on the oceanographic conditions. One are barophilic and one are barotolerant. The barophilic organism which lives in the deep sea or where the true barophiles cannot tolerate when come up out of their pressure. If you reduce the pressure, automatically you can see it will die. If anything, you could have seen anyone could have caught any fish from the deep sea by angling method. Once the fish comes, it immediately dies, not because of the temperature or the oxygen deficiency. First, it dies because of the pressure, and the full system will blot up because there is a low pressure. So to tackle this, we have developed a high pressure sampling system. It is a completely sterilizable and pressure rated system, which is rated for 600 bar pressure rated systems, which goes from the surface. This is a system which goes from the surface to the deep sea. And this systems can be triggered from the top to open it up at the specific locations where we want, because the system is controlled by the ship, which has some CTD. It is called a conductivity depth and temperature monitoring device. Based on the temperature and conductivity signals and depth signals, we can open and close up these bottles at any point of time, and we can collect the water samples without losing the temperature and contamination and pressure from the bottle. So this is the sampler of taken from the deep sea sampling, which shows the pressure difference. And once the sampler comes, immediately the task asks how to process the sample. Again, the process at the high pressure, it has to be processed without opening to the surface, without opening to the environmental pressure. So the system goes to a high pressure sampling system to a high pressure culture system, serial dilution system. Anyone from microbiology should understand the decisions to separate microbes from the water. You have to serial dilute, because every ml of the water will have 10 to the power of 12 to 10 to the power of 20 microbes per ml. So to have to isolate one, you have to several times go to the side dilution system, then achieve 1 micro. So all the system is done in the same pressure without losing even a 0.1 bar pressure. So by this, we take the samples. So we go for a high pressure sampling cultivation systems. It's a culture-based approach. And secondly, we go for a metagenome. We will compare both the things together. So one part we'll do the metagenome, that we identified cultivable and non-cultivable, and the some contents we do the cultivable high pressure. And we can compare what is cultivable, what is not cultivable, and what are the pressure tolerant and which is not the pressure tolerance. And this is a case study that we did from a deep microbial sediments. And once the sampling comes, the cultivable samples will go on high pressure, and without cultivable will go on the sequence of Illumina platform. And you should see these are the 3 samples which we have the maximum number of samples we got recently. And based on the amplicon sequencing of this sequence, we have identified at least nearly 25 phyla belonging to 54 classes and 500 genus, and phylotypes of more than 44,000 has been recorded from one single sample. That means you understand the diversity of one microbial collection which have this many phylotypes, and which is the interesting thing is not cultivable. You know, the microbial cultivations, only 1% of the general microbes we can cultivate. Less 99% we do not. So when it's on the surface, you cannot cultivate more than 1%. When in deep sea, you are not cultivating even 1%. You are cultivating 0.1%. So that itself gives 44,000 phenotypes. So you imagine what is kind of diversity available in the deep sea, which has entirely different type of mechanism to do this. And out of this, there are a variety of species which is very close to Micromonas and Saranaspora, which is very reported species from deep sea, even Mariana Trench. This species is available, and this is a phylum abundance chart, as well as in fact dominant genes. I don't want to go inside the full number and details, and we also calculated what is the operational taxonomic unit. That means active genes inside that, because only we are dealing with the genes. And we could found at least 79 type of operating genes in every single samples, every single amplifications. And what are the essence of what we did is we isolated at every sampling steps. We isolated approximately 180 isolates and 45 fungi and 123 type of actinomyces. And out of it, we were able to screen variety of compounds from actinomyces to multiple proteoglycans, griseofulvins, L-asparaginase, lipopeptides, cyclic peptides, antimicropeptides, ectoin enzymes, a huge array of chemicals which is not commercial, not available in the general microscopes seen reported there. And Apart from the technology part or the biotechnology part, we do a kind of a basic science also, how the pressure variants differentiate the organisms. So when it comes, the animal is cultured at a different organisms, the bacteria is cultured organisms, different pressure. It entirely, the expression changes, the morphology changes. You can see a single microswitch of say 3 micron in length will increase up to 15 micron in length. You can see it is completely growing like a thread, single micros. So there are interesting facts to identify what kind of termination genes is happening for this extension and reduction of size. And we have a very interesting data on this. And out of this, we have optimized variety of protocols for high pressure culture and production of biosurfactant, ectoine, and a huge number of biotechnological application compounds. So what we do after that, once we get the samples, we completely mine the microbial genome, as well as we do the microbial synthetic part, as well as do the multiple cultivation and staining part. And we try to get all those genes back or the working genes inside this and try to develop and package. For this one, we had developed and recombinant L-asparagines. You must be knowing asparagines is a very renowned chemical used for anti-cancer treatment, and it is synthesized or it is identified at least 40-50 years back and still given in a chemotherapy. L-asparagine is still known drug given in a chemotherapy, but everyone knows that the chemotherapy drugs with the cost of side effect, because it has a glycine chain built inside that, which cannot be removed. If you remove it, it will lose the activity. So that is the strain that we get. But interestingly, we found the gene and the microbes which has a gene coding which do not have a glycine coding genes. Then we get a glycine-free L-asparaginase, which means we doesn't have a side effect for doing an anti-cancer drug. So a complete gene from this BSACIST microbes has been excised, and through recombinant technology, for CRISPR way used and recombinant technology, we have transferred the complete gene platform to a working platform of E. coli, and those technologies has been utilized for a variety of things. And similarly, this is a lipopeptide. You know, the surfactant, the sense of the old detergent is surf. The compound which we call is surfactant, which is an industry synthetic chemical. And we use this, and nowadays growing interest because of that one, we want to go for biodegradable and a lot of interest on lipopeptide surfactants from microbial and so many sources. So we found a variety of organisms like Bacillus licheniformis in Andaman volcanic island activity, volcanic active islands, and we found there are having huge lipopeptide surfactant which will be used with similar technology. Same, it goes for a gene editing, then a recombinant technology transfer, and take out the genes, excised, and put inside very normal systems. And this also another one gene still and functional characteristic. Ectoine, you know, it is a commercial known pigment used for cosmetic applications and UV blocking agent. And we could able to find one gene from an bacteria Bacillus classic from deep sea samples, and the same technique it is again excised and expressed in the system. I don't want each one subject. If I say ectoine biosynthesis gene, it's itself the complete story from the microbes to this gene production is story of speaking one day. So I don't want to go inside the complete methodology inside that. I understand people can go inside that, but actually it has a lot of experimentation. It is a 2 years process to get the gene and excise and insert and do have an expression system out of that and to go for a commercial scale production. This a couple of this technologies has been transferred to industry. Already they are going for a mass scale production during fermentation using expression vectors. And this is also some kind of a beta glycine gene which is available in the deep sea cosmopod, deep sea microbes. So what is the problem in here? If anything that you use as a biotechnology product, you require the organisms exactly. You require the organism, you require the cultivator, you require to cultivate them and use to extract them. That is the only way you do all the time until present. You collect organism, either plant or animal or whatever may be, you collect it and you collect it on large scale and you extract it and take the compounds, purify that, and further go for your purification, further go for the studies. So this is one of the bottleneck in any natural systems, organic systems. So to come this is there are only one method at presently people are doing, that is retrosynthesis. So anything can be synthesized as a chemistry person must be knowing this. any compound can be synthesized. But its synthesized comes with the cost and a time, and cost and time. That means it takes number of steps, maybe 20 steps, maybe 100 steps, or the cost. Both the things will be available. To do this, this is the process. So only to do retrosynthesis of 4 amino acids, it takes 50 steps. If you take any compounds, that will be in excess of say 100 amino acids. So to synthesize that many number of amino acids, it is not so much easy. For example, we will speak about the ectasin. From it is a proven anticancer compound which is in commercial market for cancer treatment, and that is originally collected from an organism called ectasin, marine acidian. It's a soft animal. It is collected from this acidian, and this for a very long time this acidian was cultivated and extracted. For cultivation, you require a large process. You undergo the sea conditions. You have to. It is subjected by the seasonalities and natural environmental conditions, and finally the yield, and finally the compound quantity will be very less. So in later period, what has been done is they could able to identify a group of microbes which is coding for this, which is a symbiotic organism which they do produce the essential compound called ectasin. So this microbes was later on isolated, and they produced a technology to do fermentation through production. Later on, they also found the fermentation process is not stable and the genes are changing. So then they again come to a recombinant technology. Again, they have taken the genes from the microbes and do the process of producing in a specific system so that there will be no oscillations in the production. So these are some of the main natural compounds was identified and marketed from the commercial source alone, originally isolated from the rate of organism, from tunicates, from sea hair, as well as sponges and actinobacteria. So many things are coming from originally from the bacteria or the larger organism. Slowly, all the organisms has been identified, the biosynthetic pathways or the genes has been identified, and now they are in a very separate specific expression system for specific compounds. So this is the place where the biotechnology actually leads, which I spoke in a very short, but it is a very big process of optimizing and to bringing to the final scale. So this is a new concept of a drug discovery from marine biodiversity. Instead of going for diversity to finding an organism, getting the organism. Now genome mining, targeted genes, identification of secondary metabolism gene clusters, and by then you go for a new compounds by editing the genes. And this leads the way for variety of drugs from the deep sea organisms. And for sure, we know the deep sea organisms has variety of chemical pathways which a common animal or a common bacteria on the surface do not have. So this biotechnology kind of tools is being utilized, and the engineering as well as in biotechnology tool is utilized for harnessing the deep sea microbes. Thank you.
Thank you, sir. It was a wonderful lecture. Any questions? Now I request Dr. P. Rahman, the Vice Chancellor, to honor Dr. Tarani Kumar.
And I request the technicalists to give a prize to the winners. Of course, Science and Research will provide the prizes and certificate to the winners. So under the whole presentation, under this is oral presentation prizes for the on-campus only. The online will be given afterwards. So for the faculty category, Dr. D. Monisha from Tirthangal Nadar College. Faculty category. No. Okay, fine. For the research scholars category, we go Rishika Nair from BIT. Vachaga Dali from Chettinad. Anish Lupan from Bharatiya University. Abhinaya from the SRM. Abhinaya. Rashmi from Itraj College. Sureka Kattur from Sri Venkateswara Institute of Medical Sciences. Priya from SD Vaishnava College. Priya. Vaishnava. DG Vaishnava. Okay. We proceed with the student category. From Vikash from Madras Engineering College. Mohan Raj from VLRI. Prakash Krishna from Madras Engineering College. The poster presentation category, Rajalakshmi and Group from SRM. Rashmi Datta from SRM. Rashmi Datta. And the PK Kumari Singh from SRM. So I request Dr. Kathleen sir to give a words for this conference.
So actually, when we evaluate the oral presentation yesterday, we thought University of East London students are there in the BSc second year only, so we treated them as a special category. So we want for the because of the lot of efforts they put in last 40 days, so we want to award them everyone with a memento and also certificate. So maybe I'm going to call one by one. Wendy Thomas. Next, Enrica Patu. Isabel Clark. Jayashree Patel. Alicia. Sabina.
So excuse me, sir. So the name description happened. So the Priya from MGR University. Actually, quite close. So may I request Dr. Asur Masa, Associate Professor, School of Licenses, to propose vote of thanks.
Good evening, one of the present here. I know it's very late evening. I don't want to take much of your time. I'll try to finish it up as soon as possible. I would like to thank few souls who contributed for this conference as well as this tutoring program. So it's my pleasure to thank all of them one by one. First, I start with the management chairman who gave the platform for this particular tutoring grant program as well as this conference. Especially our chancellor, who is always supporting whatever the activities we do in our school. My sincere thanks goes to our Honorable Vice Chancellor, Dr. T. Mohammed, sir, who always guiding us, encouraging us, whatever the activities, especially academic and research activities conducted by School of Life Sciences. Even you can witness now, very patiently he sits and listens all the talks, every details that happens in this program. Thank you so much, sir, for your presence. I'm happy to thank our registrar, sir, Dr. Raja Hussain, sir, who inaugurated along with our Vice Chancellor for this conference and also who supported this tutoring program. Thank you so much, sir. And I would like to thank our Dean, School of Life Sciences. Anyway, on behalf of her, I'm thanking all of you. But still, it's worth mentioning. She's the backbone of this both tutoring grant program as well as this two-day international conferences. Within a short span of time, we could be able to organize very successfully, and more than 350 students we could accommodate. And around the world, the participants who presented their oral as well as poster presentations. Thank you, ma'am. Next, I would like to thank our valedictory chief guest, Dr. Dharani, who is a scientist from NIOT, for his thought-provoking talk, and also the valedictory address who gave today, present with us. Thank you so much, sir. I'm happy to thank the awardees of Turing Grant Program, who 6 people who are sitting here. I hope you all enjoyed here. That is witnessed from your feedback. Thank you so much for your presence here, and I hope you will carry some good memories from us. Thank you so much, and keep in touch with us. Thank you. I sincerely thank all the speakers from nationally and internationally who gave invited lectures as well as keynote lectures, irrespective of their busy schedules. I hope all the names were already read by Dr. Mubarak. I don't want to repeat them. Next, I would like to thank all the faculty members of School of Life Sciences because they are silently sitting back and doing all the activities, especially they gave a lot of talks and also they participate in different committees to organize this conference as well as the tutoring program. Their tireless effort must be thanked. Thank you so much. I'm extremely happy to thank Dr. Wasimalai, Assistant Professor from Chemistry. Already a lot of people mentioned his name, but still I'm happy to say thank you to you, sir, because you mentored the touring program students as well as in this conference he chaired almost like a life science faculty. He was here 2 days. Thank you so much, sir. I also want to thank our non-teaching staffs as well as Mr. Kader, who is in charge of this auditor seminar hall. Thank you so much for their tireless effort. Next, I would like to thank the coordinators of this conference, Dr. Mubarak Ali, Assistant Professor, and Dr. Ranjini, Women Scientist, who are the coordinators of this particular conference. I thank them for their contributions. Thank you. At last, the participants, without you people, this conference wouldn't be successful. Thank you so much for your patience. I know you have a little bit struggles. Still, apart from that all, you managed to stay here with this late hours also. Thank you so much. And I would like to congratulate all of you for your, the awards. Finally, I would like to thank all of you who are directly or indirectly involved in this 2-day conference program as well as Turing Grant programs. Thank you all. We will meet in the next conference. Thank you so much. Please carry good memories.
Because all the winners to come forward to do for all the faculty as well, please. So instruction to the online participants. So you'll be given the merit certificate after the assessment is over. So please stay back. It's online participants. You can continue. For photography, please stand. Come on, all the.
Not standing.
Thank you so much. See you again.
Keep put a certificate there. Here. Okay. Rasimale, Doctor Kupran. Yeah.